अंकित ये नहीं आना आ गया end stream edit and done.
यही है ना टेक्निकल सेशन 3 ऑफ इंटरनेशनल लीग कॉन्फ्रेंस आदित्य वीडियो टाइटल यही है
हाँ बराबर यस इसमें लाल लाल क्यों आ रहा है?
थंबनेल का ऑप्शन आ गया अरे तो इसमें थंबनेल का ऑप्शन नहीं आ रहा है
आएगा ना
कहाँ से आता है
एडिट पे लगा एडिट पे सर ये देखिए अभी आपके मेरे से मैं आपको मेल कर रहा हूँ टेक्निकल सेशन 1 का है 1 लिखो आ जाओगे सर ये अभी भेज दिया थर्ड का मिला
क्या
12 बजे शुरू कर देना सर 12 बजे लाइव
टेक्निकल सेशन है
हाँ
सर मैसेज बना रहे हो
सर मोबाइल सर 1 2 लिंक हमने उधर से तो फॉरवर्ड किया सर आपको ग्रुप में सुबह सब टीचर्स को हमने शेयर कर देंगे आप डाल दीजिएगा
सर हाँ क्या ये तो पेपर है ये टेक्निकल ग्रुप है ना हाँ टेक्निकल सेशन का मैसेज
मैं तो थक गया वाह फटक से बना नहीं
अरे बनाया है मेल किया मैंने
ये लिंक टेक्निकल सेशन आप उस पर टैग करके लिख देना ये सर ये अरे उसका वो मैसेज से दे देना अभी उस फैकल्टी वाले में डाला ना सेम मैसेज ऑर्गेनाइजेशन टेक्निकल सेशन 1 2 3 किधर भेजा
3 भेज सकते
अरे एक ही सेशन डाला
नहीं नो इट्स नहीं अरे नो कर दो कुछ फर्क नहीं पड़ता
बाकी सब सेम रहना है ना
हाँ सेम फटक से लिख देंगे सर इन दोनों का भेज दिया मैंने हम शुरू कर देंगे 12 बजे टेक्निकल सेशन 1
तूने स्टार्ट करके स्टॉप किया है
हाँ मैं दोनों जगह रोक दिया है मैंने जब शुरू करेंगे तब कर देंगे हाँ
यस मैम वो बोलते हैं जो प्रॉब्लम है ना क्या उसी कीबोर्ड होगा देखो 4 या 5 दिसंबर को जब मीटिंग हुआ तो उसी दिन रहेगा वो हाँ ये देखिए
इन दोनों में
सर आपने भेजा टीएसडी से टेक्निकल सेशन 3 का
उसमें चल रहा है देख आया
पॉज है यूट्यूब ओपन कर जरा
ये रहा इधर से भी मैंने रोक के रखा है
ये मैसेज परफेक्ट बन गया टेक्निकल सेशन में तीनों एक साथ रन नहीं अलग अलग
अरे स्ट्रीम एंड क्यों हो गया आदि
हाँ
स्ट्रीम एंड कैसे हो गया
अरे भाई तूने क्या किया
कुछ नहीं किया मैंने मेल ओपन किया ये रजिस्टर पे क्लिक किया
सर 2 मिनट रुक जाना
क्या हुआ टेक्निकल सेशन 1 में प्रॉब्लम है कि 2 में प्रॉब्लम
2 में
2 में 3 का हो गया
3 हो गया
3 का भेज दिया
2 मिनट सर
लिंक वही है
2 मिनट एक बार कंफर्म कर
ये आंचल थ्री का प्लॉट थ्री का हो गया।
वन का भी
2 मिनट हो जाते हैं।
अरे कीबोर्ड उस पे कनेक्टेड है। वही तो बोल रहा हूँ, टेंशन मत ले, टेंशन नहीं लेगा।
चालू तो कर देना था।
या तो इसका लिंक उस पे डाल या तो उसका लिंक इस पे डाल, दोनों की बात है। स्टार्ट कर, स्टॉप कर, उधर स्टॉप कर। अभी इसको ओपन कर।
हाँ पता देख रहा हूँ मैं, बीच में एक्सीडेंट करता हूँ।
अभी लिंक कॉपी करके अभी भेज।
कुछ फर्क नहीं पड़ेगा।
मेरे को तो सेम लिंक रहेगा सर, अगर तू जो भेजा उसको ओपन करके नहीं।
नहीं
वीडियो एंडेड दिखा रहा है।
लिंक नहीं है।
टू वाला
उधर से अभी जो 2 वाला लिंक भेजा था ना, उसके
भूल
लिए
जाइए।
अभी उसको हाँ वही भूल सही।
ये बंद कर मैंने पिछला में बंद किया कि नहीं?
हाँ किया।
अभी तक
हाँ तो वो दिखेगा क्या?
इवन आपका
फोन
फोन
की आवाज आ रही है।
दिखाई।
उसको बंद कर पहले उधर से।
अरे
रिमूव नहीं किया क्या पिछला वाला?
अरे रिमूव वीडियो रिमूव थोड़ी कर।
हाँ वो एंड हो गया था नहीं, वो लाइन में जाकर पीछे चलता है।
आधी आवाज आ रही है।
हेलो, माइक ऑफ है, सब ऑफ है सर।
तीन में किसका माइक चालू है?
इसका तो माइक है ही नहीं। कौन सा टेक्निकल सेशन हुआ? अच्छा इसका ही।
ये आदि उसका माइक बंद
नहीं वो चालू नहीं हुआ।
नहीं उधर से ओबीएस से
शमी का करते हैं।
ये वाला बंद कर उसको, ब्लू वाले से।
A very warm welcome to all of you present here, and I would like to invite you all to join our technical session number one. First, we have Ms. Vandana Sharma, whose topic is formulation and estimation of antibacterial effect of lab-made herbal soap.
I, Vandana Sharma, second year student of Microbiology ID, Thakur Shamnarayan Degree College, working under guidance with Aishwarya Kumar, is here to present my research paper on topic formulation and estimation of antibacterial effect of herbal soap. Abstract. Today's environment contains a lot of pollutions, including UV rays, which are hazardous to the human body and can damage our skin. Thus, cosmetics are an important element of skin and a body care. A herbal soap was produced by combining neem leaf, turmeric, rose petals, coconut oil, castor oil, and soap base, and it was tested against the four most common microbes found on the skin: Klebsiella, E. coli, Staphylococcus aureus, Pseudomonas aeruginosa by using herbivore method. The plant used in the soap preparation is able to soften the skin epidermis, it enhances the greater penetration, remove acne, as well as promote healing quickly. Introduction. Soap is one of the oldest hygienic products used by the people. They are important in hygiene and health because in addition to being cosmetic, they remove dirt for the human body. Herbal soap preparations are medicines or a treatment that uses chemicals that are antibacterial and antifungal and predominantly use plant parts, including leaf, stems, roots, and fruits, to treat wounds, ward off sickness, and promote wellness. The skin is a region of the body that is most exposed to the sun and environmental contaminants, which causes allergy, warts, acne, rashes on it. As a result, research is moving more sharply towards developing natural medicines that are superior to the chemical medicines in terms of quality, affordability, and side effects, which used to treat the said disorder. Herbal medicines have the benefits of being more widely available, having less side effects, and costing less than chemical treatments. To produce a quality soap, natural and handmade soap production has been a totally artistic work involving various factors such as ingredients, ability, and its employment. According to the study, choosing soap and cleaning supplies depends on how well they eliminate harmful bacteria that are frequently encountered in daily activities. Preparations of soap. The ingredients were used to make the herbal soap are coconut oil, castor oil, almond oil, vitamin E capsule, turmeric, rose petals, sodium hydroxide, glycerine, neem powder, and multani mitti. By cold process method, to make a soap base, we pour 500 ml of coconut oil into a container, place it in a water bath, and heat it to 70 degrees Celsius. The lye solution was then heated to 70 degrees Celsius and added to the coconut oil, kept at the same temperature. The mixture was thickened by adding a few petals. All ingredients were mixed well to obtain a homologous soap mixture. Finally, the soap base was poured into a circular mold with a diameter of 9 centimeters. The molds were kept aside for 3 to 4 days for solidification of soap. Method. Preparation of soap sample extract. 1 square centimeter of soap was scraped out with a strong sturdy knife. Utilizing a digital weighing scale, the weight was calculated. 4.5 ml of distilled water was used to dissolve 2.5 grams of soap sample. Isolation of microorganisms. Bacterial cultures isolated in the laboratory for the investigation included Staphylococcus aureus, E. coli, Pseudomonas aeruginosa, and Klebsiella. 4 sterile Petri plates containing neurohinton agar and 4 Petri plates with sterile nutrient agar were used. The organisms were inoculated in the plates using spread plate technique and was allowed to dry for 2 minutes so that the medium may absorb the inoculum completely. With the use of a sterile forceps, sterile filter paper discs created from the right concentration of the herbal soap sample were aseptically deposited directly on the surface of the plate. To ensure the appropriate contact and avoid the misplacement, slightly touch each disc with forceps. All the plates were incubated at 70 degrees Celsius for 24 hours before being checked for zone of inhibitions around the disc. The zone of inhibition was calculated by measuring the diameter in millimeters of the zone where the soap hide in the organism's growth. These were my observations. These were the observation table. This was the graph for nutrient agar plate and neurohinton agar plate. Result. Following incubation, an inhibitory zone was observed around some of the disc of each sample. The herbal soap demonstrated an amazing effect on different microorganisms, E. coli, Pseudomonas aeruginosa, and Klebsiella, showing an active antimicrobial properties of the soap with a minimum zone of inhibition observed on bacteria. Staphylococcus aureus with a maximum zone of inhibition observed in nutrient agar plate and minimum zone observed in neural hinton agar. Concentrated soap solution disc displayed the highest zone of inhibition in the first quadrant, whereas the diluted soap solution disc show the minimum zone of inhibition in the second, followed by third and fourth quadrant. Conclusion. Formulated soap physiochemical characteristics were satisfactory and equivalent to the control. The herbal infusion shows a notable properties including moisturizing, anti-aging, anti-acne, anti-inflammatory, and antibacterial, amongst others. These were the references. Thank you.
Thank you, Vandana. Now, I call upon Ms. Aditi Sharma, whose topic is the study of bioethanol production from expired fruit juice and its estimation using Saccharomyces cerevisiae.
Hello, I, Aditi Sharma, third year student of microbiology, I would like to present my work on study of bioethanol production from expired fruit juices and its estimation using Saccharomyces cerevisiae under the guidance of Ms. Aishwarya Kumar, Assistant Professor, Department of Microbiology, ID. Biofuels are an important factor as they substitute for fossil fuels and hence the production is increasing every day. It can be produced from feedstock, microalgae, biological remains, and energy management ensures the economic and sustainable production of protein generating biofuels. Biofuel production techniques depend upon type of raw material, efficiency level, production volume, surrounding situation, and end users' requirement. Cellulose is a polymer of glucose, which includes paper, cotton, wood, and starch food, and a simple sugar that is easily consumed by yeast to produce ethanol, which in turn can be used to produce biofuels. Substrates which contain high amounts of sugar can be used to produce bioethanol, which means fodder crops which contains simpler sugar in large amounts. In the present of various fruit juices that used to estimate ethanol production by fermentation of juice cell seeds. Juice can last from weeks to months beyond the date printed on the label since the shelf life of fruit juice depends on the variety of factors such as the type of juice, the best by the day, how the juice was stored, the packaging and actual content of the juice package. Why we chose this topic? Fossil fuels have a negative impact on the environment as green gases are emitted, which is harmful to both health as well as the environment. Ethanol is a renewable fuel made from various waste materials such as plants, collectively known as biomass. Production of biofuel from various waste plants such as agriculture waste, municipal waste, etc. can be carried out with the objective of converting the waste to useful material. Any starch or sugar waste material referred to as cellulose material which has a high amount of cellulose or sugar present in its composition, which can be converted to a fuel and it improves the energy balance of ethanol. Because cellulose feedstocks requires less fossil fuels energy to produce ethanol. Expired fruit juice, which is a waste, can be used in a bioprocessing. Bioethanol is ethanol produced from the fermentation process that can be done by various microorganisms such as Saccharomyces cerevisiae. Bioethanol is one of the most interesting biofuels due to its positive impact on the environment. Currently, it is mostly produced from sugar and starch containing raw materials. Methodology: 150 ml of sterile conical flask were taken. One conical flask were loaded with 80 ml of juice, which was bottled at 121 degrees Celsius for 30 minutes and cooled. Similarly, the other juice samples were done. After the flask was inoculated with Saccharomyces cerevisiae as a positive control. After that, the conical flasks were left in a shaker for 24 hours at 150 hours. This is a general procedure which was followed for different juice samples. The juices samples which were used are as follows. Sample 1 Tropicana pomegranate juice, Sample 2 Tropicana mixed fruit juice, Sample 3 Tropicana mango juice, Sample 4 Tropicana orange juice, Sample 5 fresh sugarcane juice. These preparation of chromic acid 400 ml, 4 gram of potassium dichromate plus 20 ml of H2SO4 plus 8 ml distilled water. Qualitative test for ethanol. First, we took one test tube for each juice sample. In the test tube, we add 2 ml of fermented broth with Saccharomyces cerevisiae, which was left in a shaker. Then 1 ml of chromic acid was added. After heating the test tube in 10 to 15 minutes at 60 degrees Celsius, if green-blue color appears, that indicates the presence of ethanol. Further, quantitative test for ethanol was done. Most of the chemical oxidation methods are based on the complete oxidation of ethanol by dichromate in the presence of sulfuric acid with the formation of acetic acid. This reaction is highly preferred because potassium dichromate is easily available in high purity and solution is indefinitely stable in type. Dichromate is yellowish in color and then reduced chromic product is intensely green. The spectra of the solution of interest must be analyzed at multiple wavelengths to calculate the individual concentration of dichromate and chromic ions in a mixture subject to the material balance that the total number of chromium atoms must be converted. Observation of fruit juice after inoculation. Juices were dull in color, they were as pungent smell like pure. Cloth of sugar degradation was observed and due to gas formation the bubbles were observed. Observation of qualitative test. Dark green color was observed in mango, orange, and fresh sugarcane juice. A light green color was observed in mixed fruit and pomegranate. As green color was observed, we performed quantitative tests for further analysis, estimation, and comparative study was done further. Estimation for ethanol from fermented media. By following the above chart, the estimation of ethanol was done for various juice samples, it was seen that after fermentation using Saccharomyces cerevisiae, Tropicana pomegranate juice produced 14.75% ethanol, Tropicana mixed fruit juice produced 15.5% ethanol, Tropicana mango juice produced 17%, Tropicana orange juice produced 23.5%, fresh sugarcane juice produced 25.5% ethanol. Readings was observed at 595 nanometers using spectrophotometer. Result and discussion. Expired fruit juice was selected on the basis of carbohydrate amount of the fruit and one fresh juice was selected to compare and analyze both of them. The selection of organisms Saccharomyces cerevisiae is done as it is ability to convert sugar into ethanol by fermentation process. Ethanol estimation was measured using chromic acid quantitative test for various samples. Highest amount of ethanol production was obtained in fresh sugarcane juice as it contains high amount of sugar which is in the form of starch. From the expired juice sample, mango and orange had the highest amount of production. Quantitative results were done in small amount from the fermented media to estimate an approximate amount of ethanol production. Further, purification and extraction of ethanol can be used in various applications. Below figure shows ethanol production in fermentation media in using orange mango mixed with pomegranate sugarcane juice as a source of carbon. Estimation of ethanol from the fermented medium. Note that this was done for 2 ml of fermented malt and not the whole fermented media was estimated. Conclusion. From the above practice, it can be concluded that as the demand of alternative fuel increases every day, production of ethanol from renewable sources such as agricultural waste, fruit waste, municipal waste, etc. is imperative for meeting increased demand. These materials could prove as cheap and abundant feedstock and have potential to produce fuel bioethanol at reasonable cost. Production of ethanol from various carbon sources is carried out by engineering or by exploiting native fermentation pathways of various microbial hosts. From this experiment, it is proved that ethanol yield could be produced from fruit juice, fresh, expired, as a substrate, and high income or profit can be obtained using material which has a high amount of sugar. Maximum activity is obtained by using mango, sugar, and orange fruit juice as the substrate at 37 degrees Celsius, as it contains high amount of starch. As mixed juice contain different number of sugars depending upon the juice, this could integrate complex sugars, and further studies with further organisms and enzyme addition as substrate can be used to fully utilize the sugar, and maybe the more amount of ethanol production can be observed. References. These are the references which were considered. Thank you.
Thank you, Aditi. Now, next we have Ms. Soni Singh, whose topic is ethanol production from barley, Hodam Bulgare, and oats, Avena sativa.
Hello, hello everyone. My name is Soni Singh, and I'm here to present my paper on the topic ethanol production from barley and oats under the guidance of Professor Ms. Aishwarya Kumar. So here we start. Introduction. Foods and beverages have long been altered by fermentation to change their flavor, texture, aroma, and microbiological stability. Barley grains is increasingly being used as a source of ethanol for gasoline. Barley is now used to make ethanol throughout Europe, but not in the US. Barley is used to make malt after being sprouted under controlled circumstances and dried in a clean. Beer is the most popular alcoholic beverage worldwide. Barley is preferable to corn for ethanol generation outside the maize belt due to a number of factors. A farmer can grow soybean sooner since barley is harvested before wheat. In many regions where maize doesn't grow well, barley might replace it as a more affordable ethanol feedstock because it does. Commercial fuel ethanol production has largely relied on two feedstocks, sugars and starch. The two nations that produce the most gasoline ethanol globally are Brazil and the US. Over the past 10 years, it has become increasingly understood that hulless barley is nutritious and may be used as functional food and has other advantages over conventional barley. Materials here used are yeast culture, that is Saccharomyces cerevisiae, inoculum medium, and fermentation medium, uh, barley, oats, and saffron broth. Few chemicals are also used for the estimation of alcohol, along with distilled water and starch indicator and the glass vessels. Methodology. Saccharomyces cerevisiae uses EMP pathway or glycolysis cycle to ferment hexose carbohydrates into ethyl alcohol and carbon dioxide. Saccharomyces cerevisiae was inoculated into a 150 ml flask containing 50 ml of sterile growth media for 48 hours of room temperature on shaker for incubation. After 48 hours of incubation, 5 grams of barley and 5 grams of oats along with Saccharomyces cerevisiae were added respectively in two different flasks and incubated at room temperature without aeration for 1 week. After 1 week of incubation by adding phenol red indicator 25% ZnS4 1N NaOH and distilled water, the mixture can be filtered and precipitate can be removed. At the end of this, we obtain a pure alcohol solution from barley as well as oats as shown in the picture. So now the methodology for estimation of alcohol by dichromate method. The dichromate technique is used to determine alcohol. Potassium alcohol is converted to acid by oxidizing agent dichromate. Isometric titration is used to measure the excess dichromate reagent once alcohol oxidation is complete. This approach is accurate for alcohol concentration between 5 and 1 and 5 mg. So after conducting the dichromate test, we can conclude our observation that in barley, the concentration of alcohol is 1.8%, and in the case of oats, it's 1.5%. So the following data are plotted on the graph. This graph is for barley and this is for oats. Result. By employing varied amount of standard alcohol, the alcohol content of fermented wheat and oat broths was estimated. Alcohol concentration ranging from 0.1 to 0.4 mg per ml were used to estimate alcohol content using fermented wheat and oat broths. and the dichromatic technique. Fermented broth was used to make alcohol. Dichromatic estimation was carried out utilizing a starch indicator and Na2S2O3 dichromate reagent. So here I would like to conclude my presentation. Thank you.
Thank you, Sumi. Now I call upon Mr. Piyush Kumawa, whose topic is comparative analysis of homemade sauerkraut made from green cabbage, Brassica oleracea var capitata, and red cabbage, that is Brassica oleracea var capita F. rubera.
Greeting to all, myself Piyush Kumawa, third year student of BSc Microbiology, studied CTC at Ashna Naik Degree College. Today I will be present my research paper on the comparative analysis of homemade sauerkraut made from the green cabbage and the red cabbage under the guidance of the Miss Aishwarya Kumari, Assistant Professor, Department of the Microbiology, ZSCTC at Thakur Shamaran Degree College. Introduction. Sauerkraut translates sour cabbage in English, which is a traditional German food. Because the fermentation process prevent bacteria from growing on the cabbage. Sauerkraut long laster than unfermented cabbage. Sauerkraut has strong unflavored and the pungent odor. Its perishes best known in the South United States. It produced by the spontaneous fermentation of cabbage, specific bacteria in the presence of 2 to 3% of the salt. The main product of fermentation is the lactic acid. Although with insignificant byproduct of fermentation, this lactic acid produces sauerkraut, its distinct flavor and the texture. Benefits of the sauerkraut. Regulates the excretory and the digestive system. Regulates the rate at which wrinkle and the age spot form. Aid in keeping a healthy heart. Increase immunity. Lower danger of the heart disease. Prevent colon cancer and the gastric ulcer. Reduce joint muscular pain and discomfort. Helpful in maintaining bone density and preventing osteoporosis. Material used for the sauerkraut preparation. Fresh cabbage, knife, container, airtight lid, salt, weighing scale. Shredded cabbage. First, this cabbage is shredded. Then add the salt, which acts as a preservative for it, which increase the number of the lactobacillus. Salt added should be 2.5% weight of the cabbage. Water comes out of the cabbage, which is called as brine. It acts as a nutritional media for the organism. Smashed cabbage is transferred in the glass jar. Keep a heavy weight on the cabbage to increase the production of the brine. For the further test, the days are first day, the 7 day, 14 day, and the 21 day. The tests are done. For pH test, the brine from the jar is tested on the first day, 7 day, 14 day, and the 21 day. The result we got is the pH decreased from first day to the 21 day by increasing the acidity of the brine due to the increase in the number of the lactobacillus. Second is the bread count. The bread count is done to determine the bacterial count in the brine. Methodology. Please slide. Make 1 centimeter by 1 centimeter square. Add 0.01 ml of brine. Spread in the square. Heat fix and stain with methylene blue. And do the further microscope. For the acidity determination, take 10 ml of brine solution. Add 10 ml of the distilled water in a 100 ml conical flask. Boil the solution to remove CO2 from it and allow it to cool. Add 5 drops of 1% phenolphthalein indicator in the flask. Try it against the 0.1 normal NaOH solution. Observe the color change from pale yellow to the pink. To find the total acidity, this is the formula. This is the observation table for the green cabbage. The physical characteristics observed were the color, odor, taste, pH, lactic acid, percent, breath count in the gram sensor. On the first day, on the second day, on the observation table of the red cabbage, we observed the result we got is many physical, chemical, and the biological changes were observed during this sauerkraut fermenting process. There was a decrease in the pH from 5.67 pH to the 3.5 within the selected time interval, which resulted in increase in the acidity order, which changed the slightly pungent to the pungent smell. The consistency changed from the hard to the soft. The taste remains salty from day 1 to the 21 days. As a result of pH change, microflora was changed from gram positive to gram negative. Number of the organisms were calculated using the breath count. It was found that the number of organisms were increased with the time. Titrable acidity of the green cabbage was found to be increased from 0.18% from first day to the 2.16% of these 21 days. Titrable acidity of the red cabbage was found to be increased from 0.45% of first day to the 2.25% of day 21. It is concluded that the acidity of the red cabbage is more than the acidity of the green cabbage. It concludes that the sauerkraut helps us study pickling process from various vegetables. Various physical, chemical, and biological changes which occur within the time interval were observed. pH and titrable acidity was checked, gram staining and breath count was performed. Study the nature, number of the microorganisms change over time, concluding that the red cabbage has higher acidity production than the green cabbage. Thank you.
Thank you. Thank you, Piyush. Next, I call upon Ms. Ashna Naik, whose topic is study of antibacterial properties of Ridol anti-ache medicine in aquarium water.
Greetings to anyone present here. I am Ms. Ashna Naik, a student of Microbiology IB working under the guidance of Ms. Aishwarya Kumar, here to represent you my research topic, study of antimicrobial properties of Ridol anti-ache in aquarium water. Introduction. In this paper, we have used Ridol anti-ache, which is used to disinfect aquarium water and maintain the health of tropical ornamental fishes. An aquarium is an artificial ecosystem that is inhabitable by various aquatic species. The environment of aquarium can differ depending upon the residing species. Beneficial microbes of the aquarium are Nitrosomonas and Nitrobacter. Pathogenic are Aeromonas, Vibrio species, Mycobacterium, etc. The antimicrobial property of this antique solution was tested during well diffusion and disk diffusion method on various agar media. The main aim of this paper is to determine whether this antique solution is also antibacterial. These are some bacterial infections. These are how fishes look when they are infected by various bacteria. Materials and method. The culture was allowed to grow on nutrient agar spread, EMB, Salmonella shigella agar, and Mullerinton agar spreads by spreading 0.1 ml of aquarium water sample on them with a spreader. The whole process is done in a sterile zone. Bells were made with coke borer. 0.5 microliter of Ridol antique was added in the wells, and the plates were incubated for 24 hours at 37 degrees Celsius. During the disk diffusion method, aquarium water was diluted to 10⁵ dilutions. 0.1 ml of each dilution was spread onto the marine agars, Salmonella shigella agars, EMB agars, and McConkey agar plates with a spreader. This was prepared by impregnating them with Ridol antique solution. Place the disc on each agar plate to check the solution's antimicrobial activity. These were incubated for 24 hours at 37 degrees Celsius, and then the zone of inhibition was calculated. These are the observations. As you can see, there's this marine agar, EMB agar plate, followed by McConkey, and then Salmonella Shigella agar. Then there is this Muller-Hinton agar plate. I have taken a photo of all the agar plates according to the serial dilution, that is from 10 raised to -1 to 10 raised to -5. These were the results. In marine agar, as you can see, for 10 raised to -1, growth and inhibition was seen. From 10 raised to -2, scarce growth was seen. And 10 raised to -3, only the scarce growth was seen, and 10 raised to -4 and 10 raised to -5, no growth was observed. Hence, there are notings found from Salmonella Shigella agar, McConkey agar, EMB agar, and Muller-Hinton agar. Conclusion. Growth on marine agar may indicate the presence of marine bacteria, that is Vibrio species, Acinetobacteria, bacteroidetes, present in aquarium water sample collected. So as growth was observed in marine agar plate and zone of inhibition was seen, we can conclude that the species that grow on marine agar were present in the aquarium water, and the antique solution can also be used to inhibit such species. By observing the zone of inhibition caused by the Ridol antake, we can conclude that these bacterial species is sensitive to antake solution, as I said before. Growth on AMB agar and McConkey agar plates indicate the presence of gram-negative bacteria, but no zone of inhibition concludes them to be resistant to the solution. Growth was also observed in EMB and McConkey agar, but there was no zone of inhibition found. Hence, we can say the gram-negative bacteria which are grown on McConkey as well as EMB agar are not inhibited by the antake solution. The absence of growth of Salmonella and Shigella agar, growth on Salmonella Shigella agar plates indicates the absence of Salmonella Shigella species in the aquarium sample. According to the principle of Salmonella Shigella agar, the growth of Salmonella and Shigella species are seen on the plates, but due to no growth observed, we can conclude that the aquarium water we collected did not have any Salmonella and Shigella species in the water. Thus, we can conclude that Ridol can be used to disinfect the aquarium tank against bacteria that are caused, bacterial infections that are caused from marine bacteria specifically. Higher the concentration of Ridol, better the disinfection. These are the references. Thank you.
Thank you, Ashna. Next, I have Ms. Aditi Sharma once again, whose topic is estimation of antibacterial properties of red sandalwood, Pterocarpus santalinus.
Hello, I, Aditi Sharma, third year student of microbiology IIT, would like to present my work on estimation of antibacterial properties of red sandalwood under the guidance of Ms. Aishwarya Kumar. Red sandalwood or Raktachanda is endemic to South India's southern eastern Ghats Mountain range. The rich red color of its wood makes this tree valuable. The wood has no aroma. Red sandalwood must not be confused with other usual sandalwoods, which is very aromatic. The products of red sandalwood can be in different forms like logs, pendants, powder, soaps, etc. As Raktachandan is used for facial benefits, we use this Raktachanda against some skin infectious bacteria and some normal flora bacteria. Environment has played a great role in spreading the resistance, mainly including factors related to soil, animal husbandry, and waste management. Resistant bacteria like Enterococcus species, Staphylococcus aureus, Klebsiella pneumonia, and etc. have been involved in causing infectious disease in humans. Raktachandan possesses some health benefits, to name some as skin pigmentation, eczema, burn, antiseptic benefits, antibacterial properties, etc. Methodology. First, a red sandalwood log collection was done. A small log of Pterocarpus santalinus was collected from a local shop in Mumbai. The wood was grated into a fine powder, which was then easily used as an aqueous extract. Preparation of aqueous extract. The red sandalwood was ground finely and then strained through a muslin cloth. A gram of sample was soaked for 2 hours in 20 ml of methanol. The sample was then centrifuged and the supernatant was picked, which served as aqueous extract for the further studies. 1 gram of sample was soaked for 2 hours in 20 ml of methanol solution. The sample was then centrifuged and the supernatant was picked, which was served as aqueous extract. By using Workman's filter paper, proper thick discs were punched and kept under UV light for 15 minutes. After this, they were submerged in paste and filtered. Agar plates. Mueller-Hilton agar plates were used for the antimicrobial susceptibility testing using Kirby-Breer dust diffusion or well diffusion method for non-fastidious bacteria, both aerobe and qualitative anaerobe. Test organism Escherichia coli, Staphylococcus aureus, Pseudomonas aeruginosa, Klebsiella pneumonia. These cultures were subculture from the master culture and were inoculated in saline suspension, and after an incubation of 1 year, the saline solution was pipetted out through a sterile 1 ml pipette. Mueller-Hilton agar plates were inoculated with the above-mentioned microorganism by spreading the suspension on the surface of media in all directions equally using a sterile glass spreader by maintaining the aseptic zone. Using a cork borer, 6 mm in diameter wells were punched into the agar as well per the Quadrant method, as well as the disc which were dipped in the aqueous extract was placed. Observation. After an incubation, the zone of inhibition was observed around the disc sample, which was dipped in the methanol aqueous solution of Raktachandran. Against Staphylococcus aureus, Raktachandran showed a resistance in higher amount as compared to other organisms. These are the observations of the zone of inhibition which was observed against the organism. Escherichia coli can cause skin infections such as cellulitis and on rare occasions in type 1 necrotizing soft tissue infections, where layers within dermis, subcutaneous tissue, superficial fascia, or muscle become infected. Staphylococcus aureus cause the majority of skin and soft tissues infection. It colonizes skin in 20 to 30%. Pseudomonas aeruginosa is most frequently associated with opportunistic infection. Klebsiella pneumoniae enters a break in the skin. It can lead to a skin or soft tissue infection. By this, we can say that the skin infectious organisms were the growth of them was inhibited by this Raktachandra. Result and conclusion. The study of the Raktachandran was carried out to determine the antibacterial properties it carries. According to the result, we can see that Raktachandran showed a zone of inhibition, that is, the growth of the bacteria can be inhibited by the Raktachandran against these test organisms which are usually present on our skin or can cause deadly or infectious infection. The test organism can cause mild to severe infections to humans depending upon dosage, and application of Raktachandran can further lead to use to treatment of normal skin infections like pimples, cysts, allergy, etc. It can be used as antibacterial cream or product in vast range of variety, which will not only act as a barrier against the bacteria or normal flow of the skin, but also can be a preventive measure towards adverse side effects of chemical products. References. These were the references which were considered. Thank you. I would like to thank my college, Zabru Singh Charitable Trust, Thakur Shamnaran Degree College, and my principal, Dr. Saraswarari Sir, also my coordinator, Mr. Udayban Yadav Sir, and my faculties, Ms. Sonali Joshi Ma'am, Ms. Niyati Pandey Ma'am, Mrs. Bharti Wadekar, Mr. Kunal Thakur Sir. Thank you.
Thank you, Aditi. Next, we have Ms. Priyanka Balika, whose topic is antioxidant and antimicrobial activities of garlic, allium sativa, and ginger officinale extract.
My name is Priyanka Gaurikar from student of second year microbiology ID from Thakur Shamnaran Degree College under the guidance of Aishwarya Kumar. My topic is antioxidant and antimicrobial garlic and ginger extract. Introduction. Antioxidants are natural substances that prevent or delay of some types of cell damage. Antimicrobial substances are materials that have activity against bacteria. Medicinal plants have been a source of novel drug compounds. Products derived from plants have been made a large contribution for human health. Two such medicinal plants are ginger and garlic. Garlic belongs to family Alliaceae. Is a potential inhibitor for pathogen. The use of garlic can increase shelf life and decrease the possibilities of food poisoning and spoilage of processed food. Ginger plays an important role in prevention of diseases. Its constituents shows vital role as anti-inflammatory process. Ginger shows antimicrobial property against growth of bacteria. Natural species of garlic and ginger possesses effective antimicrobial activity against multi-drug clinical pathogens and can be used for prevention of drug-resistant microbial diseases. Antimicrobial activities of garlic and ginger will be determined using different microorganisms. For this research, the most common bacteria, E. coli, is considered. Materials and method. Preparation of extract. Ginger and garlic was peeled, washed, finely chopped into small pieces, and paste was made, and its juice was extracted. There were 7 samples. Fresh ginger paste, 3 days of ginger paste, fresh garlic paste, 3 days old garlic paste, fresh ginger and garlic mixed paste, 3 days old ginger and garlic mixed paste, and market ginger garlic paste. In petri plate, the sterile Mueller-Hinton agar was placed and the Escherichia coli was spread by the spreader. Antimicrobial disc was dipped in the juice extracted from the sample, and it was kept for 37 degrees Celsius in incubator for 24 hours. This method for antioxidant property of ginger and garlic, the determination of antioxidant activity 2,2 diphenyl 1 picaryl hydrazine radial stanzing method was also be used. DPPH is most widely used in antioxidant assay. The free radical scavenging effect of all the extracts with different concentration was assayed by the decolorization of ethanol solution of DPPH. DPPH gets reduced in front of antioxidants. These were my observations of ginger garlic paste of 7 samples, the antibacterial activity of ginger and garlic, and its comparative test with ginger garlic paste and market paste was determined. The zone of inhibition is response to concentrated ginger and garlic extract was seen against Escherichia coli. These are the zone of inhibition diameter where fresh garlic and fresh ginger are more antioxidant property than 3 days old ginger and garlic and compared to the market ginger garlic paste. This means the fresh ginger garlic shows antioxidant properties than others and it is good for our health. This observation of for DPPH method where antioxidant samples were dipped into the DPPH reagent where the color turns to colorless or yellow from purple. I conclude my research paper. This study shows how natural and fresh items can help to lessen the harm that antibiotic resistant bacteria represent to human health. It is therefore recommended that further studies be carried out on the efficiency of garlic and ginger extract to enhance the primary healthcare delivery system. This study indicates that species like garlic and ginger have antimicrobial and antioxidant properties. In this study, the antioxidant activity of this selected species is indicated by higher antioxidant index as determined by DPPH method. This assay has the ability to measure the extract by donating hydrogen to the DPPH radial, resulting in bleaching of the DPPH solution. Spices are a rich source of polyphenolic compounds having strong antioxidant capacities and could potentially replace the synthetic antioxidant in food system and offer an additional health benefits. Consumption of spices have been implicated in the prevention of many chronic diseases such as cardiovascular diseases, cancer and inflammation, etc. In our present studies, different antioxidant capacities was observed related to different extracts of the species, and the activities were comparable to the well-known antioxidant and were able to inhibit the growth of E. coli, indicating how it can also help in increasing the gut health. This was the reference of research paper where I have references. Thank you.
Now we have Ms. Aditi Sharma once again with the topic a comprehensive study and analysis on usage, leaking, acceptance, safety, and accessibility of menstrual cups.
Hello, I, Aditi Sharma, third-year student of microbiology ID, would like to present my work on a comprehensive study, analysis, uses, leaking, acceptance, safety, and accessibility of menstrual cup under the guidance of Ms. Aishwarya Kumar, Assistant Professor, Department of Microbiology, ID. Menstrual products for females must be efficient, secure, and reasonably priced. Menstruation is a typically body process and an indication of sound reproductive function. There aren't many options for managing menstruation. Misinformation, description, high expenses, safety concerns, and cultural taboos can prevent women from exploring all the options. A lack of accessible and efficient products can cause girls and women who are menstruating to face consequences which can affect their health. The commonly used menstrual products are sanitary napkins, tampons, previous underwear, or clothes which are sometimes coated with chemicals which can be harmful for long term. One such product is the menstrual cup. A menstrual cup is a tiny silicone receptacle developed for medicine and used to insert it into the vagina to collect the menstrual flow. Silicone cups have no additives and are hypoallergenic. A single cup has a lifespan of about 10 years and is discreetly portable in pocket or purse. Menstrual cup is a reusable product. As we know, microorganisms are everywhere around us. They need favorable conditions to grow. Urinary tract infection is a microbial infection in the urinary tract of women, usually common in women when they come in contact with any of the bacteria due to the following diseases: unhygienic washroom uses, frequent contact of vagina with an unhealthy climate, not changing undergarments, not cleaning seats before urinating, not cleaning and washing the vagina properly, not changing sanitary pads, using an unclean menstrual cup. Urinary tract infections, suspected etiologies are Escherichia coli, Staphylococcus aureus, Proteus species, Klebsiella pneumoniae, species, Pseudomonas aeruginosa, and etc. These all increase health risks and there are chances of painful urination, abdominal cramp, bloating, recently toxic shock syndrome, PSS, etc. Depending upon the microorganisms and its toxicity, the symptoms may vary. Why we choose this product? To give them a fresh and sterile appearance, sanitary pad features fibers that have undergone chlorine bleaching, dioxin, a harmful contaminant produced during this bleaching process, has been linked to endometriosis, cancer, pelvic inflammatory disease, and organ disruption, causing the use of napkins to be unhealthy and to the great risk to women. The main objective of this topic was to understand the types of female menstrual cycle and issues faced by them during the menstrual product uses. There is a various way to deal with menstrual cycle. Basically, menstrual cup is one such product which is simply modern choice which is made by women. I can imagine more than 100 responses were gathered. Few Google Forms also, which not only clarified the uses but also reviewed the cultural values and certain issues which stopped women from using the menstrual cup. As being the growing generation, we are leading to choose a better and easier way to live as a generation for a simpler and safer approach to deal with the monthly menstrual cycle is a menstrual cup. The reusable menstrual cup is an alternative to a messy and shaky menstrual cycle or not. The increased collecting inside the cup cannot be felt and wearers set comfort and sense of freedom. Used to be survey formed to gather information from young girls, teenagers, adult women who are menstruating and are 12 years or older or younger. To learn how much women are concerned about their menstrual cup and what preferences they have, a series of questionnaires were age group then. This graph right to the unit shows that when they were asked about the question, how they, at what age they were aware about the menstrual cup, 13 to 18 age group, that is the 70% of the data were aware of the menstrual cup at the age group of 13 to 18 years, and most of them were unaware. Sterilization methods. When they were asked about the sterilization methods for normal purpose menstrual cup can be sterilized in the following ways: infection-free for the next use, normal water wash, hot water bath, normal water with soap, hot water with soap, sterilization machine. Out of 100 women, 60% prefer to choose a boiling water bath and 20% choose a sterilization machine. When they were asked about why did you decide to try a menstrual cup for the first time, if you haven't tried one, what is it stopping you from? Due to unawareness and a lack of knowledge, many of the females chose a sanitary pad as the lifesaver, even though it takes a lot of hygiene to maintain. A proper knowledge and trial can help them choose a better product with ease and love. Like this, we can say we all still face cultural taboo in India, which leads to misconception about the product. Result and discussion. By the survey research, we can say that many women are still facing problems while using a menstrual cup or any other menstrual product. It is a fear and lack of knowledge. The women who barely use a menstrual cup are happy with the results and also they recommend it to other women. Most of the women were unaware of menstrual cup but were facing issues with using it and not choosing it. Many of them had a fear of using it before even trying it out. Till now, there is no external health issues or case have been recognized in the use of menstrual cup. In India, 25% of women from all over the world have started to use this product. Our review indicates the menstrual cups are safe option for menstruation management and are being used internationally. The cups are not only cheap but also safe for the environment. But further studies are required to get to know the antimicrobial properties which will help us to maintain good hygiene and less urinary tract infection or any other health risks which were faced during menstruation. Good quality studies in this field are needed. Further studies are needed on antimicrobial effects of different menstrual cups and aftercare. As you can observe, more women were familiar with the products, but half of them used it and half of them not. With choosing an alternative form of menstrual cup, you need proper knowledge of using it and proper knowledge of handling it. Sanitary pads are made up of plastic, and research has said using pads and tampons fill the equivalent of 2 minutes with waste, which will take more than 100 years to decompose. A better product with not only comfort but also no other heritage should be chosen to maintain a quick in total an estimated 500 million lakh access to menstrual pads and integrated facilities for menstrual hygiene management and nurture. To effectively manage their menstruation, girls and women require access to water, sanitation, and hygiene wash facilities, affordable and appropriate menstrual hygiene materials, information on good practices, and supportive environment where they can manage menstruation without embarrassment or stigma or without any great health risk. Conclusion. Despite the limitations of this study, it demonstrates that the menstrual cups can replace traditional menstrual situation method due to lower cost and improved hygiene. Increasing awareness and accessibility to this product can aid in its adoption. Government agencies can increase the assistance of female health workers to raising awareness and facilitate the use of this device, thereby improving menstrual hygiene of the workers, which is critical in developing countries such as India. It could be concluded that menstrual cups are a more environmentally friendly option than other uses vaginal devices, which has disposable issues. Adequate secure sanitation and menstrual hygiene products are not only a part of the solution of women's health issues. We conclude that the menstrual cups need further studies and detailed knowledge for more further use. However, until now, it has been a safe choice and known to women's menstrual health. Through social media, content spread awareness towards this product, and in developing countries like India, its natural products are free of cost and 1 woman can contribute and save the environment by 1 person because yearly uses of sanitary pad is equal to no amount of degrading capability. These are the references which were considered by writing the paper. Thank you.
Thank you once again, Aditi. Next, I have we have Ms. Anuksha Jadhav, whose topic is on comparative study of anti-acne creams and serum against lab-made herbal soap on acne.
Hello, myself Anuksha Jadhav, student of ZSCT Thakur Sham Narayan Degree College, working under guidance of Aishwarya Kumar. Today, my research topic is comparative study of anti-acne cream and serum against lab-made herbal soap on acne. Abstract: the use of herbs on skin disorder has been done from thousands of years. Herbal remedies and cream, including those for many kinds of disorder, especially skin disorder, are currently gaining popularity among patients. In Asia, especially in Southeast Asian countries, herbal treatments that have been used from ages are now being studied scientifically, especially the system of Ayurvedic medicine combined with physiological and holistic principles. Acne, also known as acne vulgaris, is a long-term skin condition that occurs when dead skin cells and oil from the skin clogs hair follicles. Typical features of this condition include blackheads or whiteheads, pimple, oily skin, and possible scarring. It primarily affects the skin with a relatively high numbers of oil glands, including the face, upper part of the chest, and back. Susceptibility to acne is a primarily genetic in 80% of cases. Many different treatments exist for acne. Thus include alpha hydroxyl acid, antihydrogen medication, antibiotics medication, azelaic acid, benzoyl peroxide, hormonal treatment, etc. Acne treatment work in at least 4 different ways, which are the following: reducing inflammation, hormonal manipulation, killing C. acnes, and normalizing skin cells, shedding and sebum production in the pore to prevent blockage. Introduction: acne vulgaris is also known as acne in a chronic skin disease. Acne vulgaris is the formation of comedones, papules, pustules, nodules, and or cyst can needles on obstruction and inflammation. Treatment in based on severity can involve a variety of topical and systemic agents directed at a reducing sebum production, formation, inflammation, and bacterial count and at a normalizing keratinization. Acne vulgaris has been associated with Staphylococcus aureus, propionic bacterium acne, substitutional in vitro. Research shows that S. aureus may have a pathophysiological involvement in acne vulgaris. Both S. dermis and P. acne were identified as bacteria likely to cause acne vulgaris. According to Ayurveda, the major cause of acne are impurities present in the body. An acne-removing herbal soap comprises an alkaline solution, oil and fats, and acne-removing additive, a herbal additive. And it is characterized in that the acne-removing additive comprises aloe juice, honeysuckles, and white chrysanthemum flowers, and the oil and fats comprises coconut oil, plum oil, olive oil, cocoa butter, sesame oil, and beeswax. Material and methodology: saline suspension of acne vulgaris. Dilution of acne vulgaris was made in sterile saline tube. B. Collectively of collection of 4 different antimicrobial product sample. Sample 1 is cleaning gel. Sample 2 is vitamin C serum 20%. Sample 3 lab made herbal soap made out of coconut oil, neem, turmeric, and vitamin E. Sample 4 was face wash. C. Isolation and enrichment of sample. Cotton swab of Acne vulgaris was spread on 4 mH agar plate and 4 nutrient agar plate, and sterile disc which has been soaked in the sample and placed on the plates of Kirby-Boer method. D. Incubate the plate for 24 hours in 37 degrees Celsius. Measure the zone of inhibition caused by all the 4 samples of the same bacteria of Acne vulgaris. From this, we have found that natural soap is better for skin than chemical cosmetics. Here are some observation pictures. Sample 1 containing cleaning gel of Na and MH agar plate. Sample 2 vitamin C serum 20% of Na and MH agar plate. Sample 3 lab made herbal soap of Na and MH agar plate. And sample 4 containing face wash of Na and MH agar plate. Results. After the incubation of 24 hours in 37 degrees Celsius, inhibitory zone were observed. We observed that the 3 samples, that is cleaning gel, vitamin serum 20% and face wash, showed a minimum zone of inhibition, while a greater zone of inhibition is seen in the herbal soap sample against Acne vulgaris, indicating that the formulated soap physiochemical characterization was satisfactory and equivalent to the control. The herbal infusion showed notable properties including moisturizing, anti-aging, antibacterial, among others. Conclusion. While concluding, we can say that the herbal soap is better zone of inhibition against Acne vulgaris bacteria compared to the marketed product safe to be effective against acne. Thank you.
Thank you, Anand. Now I call upon Ms. Madalasa Milan Patel, whose topic is the use of natural pigments of red beetroot, formacid, beta vulgaris L, to develop a mycological stain and eco-friendly alternative substitute.
I, Madalsa Milan Patil, would like to give a presentation on use of natural pigments of red beetroot pomace beta vulgaris L to develop a mycological stain and eco-friendly alternative substitute under the guidance of Niyati Tiwari ma'am. Let's start with the introduction. The red beetroot, scientifically called beta vulgaris L, is a good source of natural stain. It produces red and yellow pigments known as beta-lanes. Beta-lanes consist of beta-sines, which produces red pigment, and beta-xanthins, which produces yellow pigment. The aim of the study of this experiment was to develop a mycological staining reagent from the red pigment extracted from the beetroot pomace. The availability of botanical products today has generally greatly increased consumer performances for using natural dyes to minimize the health hazards of synthetic and inorganic dyes. Staining processes are an important requirement for microbiological laboratories for the testing and identification of microorganisms containing various inorganic synthetic reagents. Therefore, plant products can provide a natural biodegradable resource for such chromogenic pigments. Materials and methodology. First step is collection of plant material. Second step is extraction of crude natural dye. Third step is purification and preparation of the stain. Fourth step is preparation of test cultures. Fifth step is staining procedure and microscopy. Sixth and the last step is agar-well diffusion method. Some of the antibacterial and antifungal properties of beetroot are as follows. Beetroot is a plant with natural anti-anemic, antibacterial, anti-carcinogenic, antipyretic, antioxidant, anti-sclerotic, detoxifying, and diuretic properties. This medicinal plant has been used medicinally for 2000 years. Packed with essential nutrients, beets are an excellent source of fiber, folic acid, which is vitamin B9, manganese, potassium, iron, and vitamin C. Increase pressure, exercise performance. Studies have also confirmed that beetroot extract has antimicrobial properties that can provide protection against Escherichia coli, Pseudomonas aeruginosa, Staphylococcus aureus, and Bacillus cereus. Results are as shown in the picture. No antibacterial property is seen and antifungal property has been seen. Antibacterial activity. The extract of beta vulgaris L did not show antibacterial effect on E. coli by performing well diffusion method. Antifungal activity. The extract of beta vulgaris L showed staining effect on yeast. Conclusion. Extract of beta vulgaris L were evaluated for the presence of antibacterial and antifungal activity. The extract of beta vulgaris L can be used as eco-friendly stain for staining process. These are some references which I have used for making my research paper. Thank you.
Thank you so much, Madrasa. Next, we have Ms. Tanvi Mene, whose topic is phytochemical and antibacterial analysis of ethanolic extract of leaves of Clitoria ternatia.
Hello everyone, I Tanvi Mene would like to give quick presentation on antibacterial and antifungal analysis of ethanolic extract of leaves of Clitoria ternate under the guidance of Niyati Tiwari ma'am. Starting with the introduction, many people are aware that eating plant-based foods add much needed fiber, vitamins and minerals to the diet, but what is less well known is the many benefits of the phytochemicals. Phytochemicals are non-nutritive chemicals which possess protective or disease-preventive properties. Clitoria ternate Lin, family Fabaceae, is a herb commonly found in India, China, Philippines, and Madagascar. It is commonly called Shankhapushpi. In traditional Ayurvedic medicine, it has been used for centuries as a memory enhancer, anti-stress, antidepressant, anticonvulsant, tranquilizing, and sedative agent. The root extract are capable of curing whooping cough. Materials and methods. Preparation of ethanol extract. Blue flowered leaves of Clitoria ternate were collected and washed with distilled water. Leaves were shade-dried and stored in airtight containers. The shade-dried leaves were coarsely powdered by mechanical grinder. The dried powder samples were extracted with 70% ethanol in the Soxhlet extractor. Extraction process was continued until the color of the final drop of the extracts became colorless. The extracts so obtained stored in airtight bottles at 4 degrees Celsius. Antimicrobial assay. Escherichia coli was used to determine the antibacterial activity and Saccharomyces cerevisiae was used to determine the antifungal activity. Antimicrobial activity and antifungal activity was determined by disk diffusion method. Nutrient agar was used to determine the antimicrobial activity. And saprolite dextrose agar was used for antifungal activity. Suspension of test organisms were made in saline. The paper discs saturated with the extract of Clitoria ternate were placed on the lawn of bacteria seeded on the surface of agar plate. The plates were incubated at 37 degrees Celsius in incubator for 24 hours. Here are the results of antibacterial activity and antifungal activity. Antibacterial activity, the extract of blue-flowered Clitoria ternate leaves did not show the zone of inhibition for Escherichia coli. Antifungal activity, The extract of blue-flowered Clitoria ternate leaves showed the zone of inhibition. The extract was effective against Saccharomyces cerevisiae. The conclusion: ethanol extract of blue-flowered leaves of Clitoria ternate were evaluated for the presence of antimicrobial activity. The presence of various bioactive compounds indicates that these leaves extract compounds can be used as therapeutic drugs for skin disease, asthma, burning sensation, and hair migraines. Here are some references I used for the research paper. Thank you.
Thank you so much, Tanvi. Next, I call upon Ms. Mansi Patel, whose title is To Study the Effect of Rita Herb Anti-Dandruff Shampoo and Comparison with Marketed Shampoos.
That I, Mansi Patel, would like to give a presentation on the topic to study the effect of Rita Herb Anti-Dandruff Shampoo and comparison with marketed shampoo under the guidance of Niyati Tiwari ma'am. To start with the introduction, hair is one of the vital parts of the body derived from ectoderm of the skin and its protective appendages on the body and considered accessory structure of instrument along the sebaceous gland, sweat glands and nails. They are also known as epidermal derivatives as they originate from epidermis during embryological development. Shampooing is one of the most common form of hair treatment. The primary function of shampoo is aimed at cleansing of hair necessitated due to accumulated sebum, dust, scalp debris, etc. The aim also includes to eliminate alkanamides material from shampoo and to put effort on formulation of natural anti-dandruff shampoos, their evaluation and comparison with marketed shampoos. In present work, Sependus mucurus vita is used to formulate the anti-dandruff shampoo and comparatively evaluate the anti-dandruff activity in combination and with the marketed anti-dandruff shampoo. Evaluation test parameters. Evaluation is very necessary to assess the quality of formulated herbal anti-dandruff shampoo. Mainly two different types of evaluation has been done, namely physiochemical evaluation and anti-dandruff activity evaluation. To start with physiochemical evaluation, first is foam formation. Shake test. About 50 ml of 2% solution of formulated anti-dandruff shampoo were taken in the 10 ml measuring cylinder and shake the cylinder about 10 times. Foam retention. After the foam formation from the shake test, reduction time of foam settlement was recorded. Cleaning action. By the Burnett and Powers method, the formulated anti-dandruff shampoo were evaluated to assess the anti-dandruff activity. Cleaning action was then recorded. Total solids. In the tarred evaporated dish, accurately weighted quantity of shampoo 4 grams is placed. Shampoo was evaporated at as low temperature as possible until the solvents were removed. The weight of the shampoo solids after drying was recorded. Determination of pH. Both the shampoos were dipped in the pH paper. The pH of both the shampoos were recorded. Second is anti-dandruff activity evaluation. Preparation of pre-inoculum. Take a loop pool of culture of Saccharomyces cerevisiae aseptically and transfer it to the sterile and cool 25 ml of Sabrod dextrose broth and mix well. Incubate the broth at 25 degrees Celsius for 24 hours. Preparation of 4 plates. A Sabrod agar 150 ml is autoclaved and poured to the already autoclaved plate and cooled in room temperature and allow the setting. By the help of sterilized cotton, take a culture of Saccharomyces cerevisiae and aseptically Swab on the agar surface. Disc diffusion method. The paper disc saturated with both the shampoos were placed on the lawn of fungus S. cerevisiae, seeded on the surface of Sabrod dextrose agar plate. The plates are incubated at 37 degrees Celsius in incubator for 24 hours, and the zone of inhibition was measured to determine the antifungal activities. Here are the results of foam formation and pH test. As you can see in the picture, there is a zone of inhibition in the herbal shampoo and no zone of inhibition in marketed shampoo. Here is the observation table and the result. The foam formation in both the shampoos are positive. Foam retention in a formulated herb anti-dandruff shampoo was 45 minutes, and in the marketed shampoo was 1 hour 30 minutes. Cleaning action, the formulated herbal anti-dandruff shampoo was easy to clean. The marketed shampoo was taking time to clean. Total solids were 0.6 gram and 1 gram. pH of the shampoos were 6 and 3 respectively. Conclusion. Conclusion can be made from the present study that herb used for preparation of anti-dandruff shampoos such as Rita possesses considerably anti-dandruff activity. Rita possesses more antifungal activity. Here are some references which I used to make the research paper. Thank you.
Thank you so much, Mansi. Next, we have Ms. Lakshata Gohil, whose topic is on the study of herbal skin protectant and its antimicrobial properties of aloe vera.
Hello everyone, myself Lakshata Gohil would like to give presentation on study of herbal skin protectant and its antimicrobial properties of aloe vera, working under the guidance of Ms. Niyati Tiwari ma'am. Firstly, we'll see the introduction. The botanical name of aloe vera is Aloe barbadensis Miller. Aloe vera is a succulent plant with thick, fleshy, lanceolate shaped leaves of green-grayish color. Aloe vera inner gel is obtained from the lower leaves of the plant by slicing the leaves open. The gel is clear, odorless, and tasteless, and should be free of leaf skin of yellow parts. The skin absorbs aloe vera up to 4 times faster than water. It appears to help pores of the skin open and receive moisture and nutrients of the plant. Aloe vera gel was studied for the microbial activity against the gram-positive and gram-negative bacteria using this diffusion method. The extract shows varied levels of antimicrobial activity against the test pathogens. So here we have materials and methods. Aloe vera leaves were collected from the plant tree. Test organism used Escherichia coli and Staphylococcus aureus. Preparation of aloe vera gel extract. The fully expanded leaves of aloe vera were selected from the plant. It was washed in distilled water and was subjected to surface sterilization with 70% of ethyl alcohol followed by 0.1% of HgCl2. The parenchymatous covering of the leaves were peeled and the gel drained out. Slurry was formed. Preparation of the extract. For the preparation of ethanol and methanol extracts, fresh leaf gel was dried in the incubator at 80 degrees Celsius for 48 hours. Then the powder, 20 grams of the powder was soaked in 200 ml of the solvent, ethanol and methanol for 24 hours. The contents then filtered through Whatman filter paper 1 and the filtrate was evaporated to dryness. The dried extract was further powdered and then dissolved in distilled water. Acetone extract was prepared in a similar manner, except that the extracted powder was dissolved in 0.15 N NaOH and further neutralized with 0.15 NaCl. So here we can see antimicrobial activity of the aloe vera gel extract. Antimicrobial activity of aloe vera gel extract was done by using this diffusion method. The paper of disc saturated with the extract of aloe vera were placed on the lawn of bacteria seeded on the surface of agar plate. The plates were incubated at 37 degrees Celsius in the incubator for 24 hours, and the zone of inhibition was measured to determine the antibacterial activity. So here we can see the comparative results of the homemade aloe vera gel extract and the marketed herbal gel. So the antibacterial activity of aloe vera gel extract against the selected bacteria, we can see zone of inhibition was observed in the mixed culture of S. aureus and E. coli in the homemade aloe vera gel extract, and no zone of inhibition was observed in outside aloe vera gel extract. So here we concluded that the aloe vera is a commercial important medicinal plant, and different parts of its fleshy leaf, that is the gel, latex, and the whole leaf extract, are widely used in the variety of forms in the food, cosmetic, and pharmaceutical industries. Aloe vera gel and whole leaf materials did not show any vitro cytotoxic effects. Here are the references which I took to prepare my presentation.
Thank you so much, Lakshmita. Next, I call upon Ms. Santa Pawar, whose title is Phytochemical Analysis, Antimicrobial Activity and Antifungal Activity of Pomegranate, Tunica grantanum L, and Plant Extract Against Microbes.
Hello, my name is Sanika Kumar. I am from Aishwarya Microbiology from Thakur Shamnara Degree College. Here I am presenting phytochemical analysis, antimicrobial activity, and antifungal activity of pomegranate and plant extract against microbes, working under the guidance of nearly Ivari Lab. Introduction. Pomegranate is one of the oldest edible fruits, widely grown in the parts of Asia, North Africa, around the Mediterranean areas, and in the Middle East. The pomegranate is a symbol of life, longevity, health, morality, immorality, and spirituality. Pomegranate exhibits antiviral, antioxidant, anti-cancer, anti-proliferative activities. The phytochemistry of pomegranate has been widely studied, and this fruit is found to be the earliest source of polyphenolic compounds. Both flavonoids and tannins are more abundant in the peels. Peels of pomegranate contain a wide variety of phytochemical compounds like tannins, ellagic acid, gallic acid, unic allergens, etc. Materials and methodology. Plant sampling. The pomegranate fruits were collected from the local market of Andhra area, Mumbai, Maharashtra. The English scientific and family names of the plant under the study are pomegranate, Tunica grantum L, and lychracea, respectively. Preparations of crude pomegranate peel. Peels were cut into small pieces, placed into plastic containers, and were kept in a freezer at -4 degrees Celsius for at least 16 hours. The frozen peels were freeze-dried. For 96 hours using freeze-dried. Dried peels were then stored in a desiccator for 1 week before being ground into powder. Preparations of extract. Dried pomegranate peels were ground into powder using a grinder at 9676.8 grams. Powders were kept in airtight plastic containers and stored at -40 degrees Celsius under use for extraction in Foxler apparatus in 250 ml of ethyl acetate. Ethyl acetate was added to 0.5 gram of dried sample in oil flask and was steamed for 3 hours at room temperature, 20 degrees Celsius. Ethyl acetate extract was capable of recovering many compounds, thus ethyl acetate was chosen for the extraction of functional components from pomegranate peels. The extraction was performed for 48 hours and concentrated by slow evaporation process. The obtained extract was kept in moisture-free container and used for phytochemical analysis. To avoid light exposure, the flask was covered with aluminum foil. Mixture was then centrifuged for 30 minutes at 976.8 gram at 3 degrees Celsius. The supernatant was filtered using Wattman filter number 1, 155 mm. Extract was then stored at 4 degrees Celsius until used for analysis. Qualitative phytochemical screening methods. Pomegranate peels were screened for the presence of key families of phytochemicals. The presence of sterols were detected by Swarovski's test respectively. The occurrence of phenolic compounds and tannins confirmed by ferrichloride test respectively. The flavonoids were detected by lead acetate test. Phytochemical investigation. Various phytochemical investigations were performed using obtained extracts. The procedures are already reported by number of workers used without any modification. The phytochemical name and investigation methods are as follows. Phenols. To test the phenol presence in a test tube, 1 ml of extract and 2 ml of distilled water were added, followed by few drops of 10% ferric chloride. Appearance of blue or green color indicates presence of phenols. Flavonoids. To test the flavonoids presence in a test tube, 1 ml of extract, a few drops of dilute sodium hydroxide was added. An intense yellow oil was produced in the plant extract which becomes colorless on addition of few drops of dilute X acid indicates the presence of flavonoid. Tannins. To test the tannin presence in a test tube, 1 ml of 5% ferric chloride was added to solvent-free extract. The presence of tannin is indicated by the formation of bluish black or greenish black precipitate. Steroids. To test the presence of steroid, 1 ml of extract was dissolved in 10 ml chloroform and equal volume of concentrated sulfuric acid added by the size of test tubes. The upper layer turns red and sulfuric acid layer shown yellow with green fluorescence. This indicates the presence of steroids. Antimicrobial activity. The plant species were studied to evaluate their antibacterial against food poisoning, microorganisms including 2 strains of gram-negative bacteria, that is gram-positive bacteria subtilis and S. aureus. Antimicrobial activity. The plant species was studied to evaluate their antimicrobial against food poisoning microorganisms, including 2 strains of gram-positive bacteria, Bacillus subtilis and Staphylococcus aureus, and 1 strain of gram-negative bacteria, Escherichia coli, using risk diffusion method and agar-well diffusion method. The results revealed that both plant extracts were effective in suppressing microbial growth of food poisoning bacteria with variable potency. Pomegranate was the most effective extract in targeting microbial growth of all tested pathogenic bacteria at concentration of 10 mg/ml. Pomegranate extract were the most effective extracts and showed a strong antimicrobial activity against food poisoning microbes. In general, gram-negative bacteria is less sensitive than gram-positive bacteria. The reason for less sensitivity of the gram-negative bacteria than gram-positive bacteria would be for the difference in cell membrane compounds and arrangements or their outer phospholipid membrane and prions constituent. Procedure disk diffusion method. This is procedure for disk diffusion method. We need sterile antimicrobial disk and also sterile petri plate. Sterile disk is placed on the sterile plate. Then the plant extract is taken in known volume by pipetting, and then the plant extract is added over the disk for impregnation to occur. In this method, if plant extract is added on into disc, then the disc will absorb the extract and then it is placed onto the media along with the culture to screen for antimicrobial activity. After that, in inoculated sterile nutrient about pH 0.2 ml of the plant extract is prepared and spread all over the plate by spread plate method. Then the sterile disc in which we have inoculated plant extract, it is taken and placed into the center of the media by giving a gentle press, and it is kept for 4 to 5 days. Antifungal activity. Antifungal activities were evaluated using plant extract against Aspergillus niger using well diffusion method. The culture of 48 hours old brood or loose bevel agar were used for inoculation of fungal strain or several dextrose agar plates. Suspension of fungal spore were prepared in sterile phosphate buffer saline. An liquid of inoculum was introduced to several dextrose agar and poured into a dish by pour plate method. After solidification, the appropriate wells were made on several dextrose agar plate by using sterile or pour. In agar well diffusion method, 1.01 ml of ethanol and acetone extract of pomegranate introduced. The zone of inhibition around the disc was measured after 24 hours of incubation at 37 degrees Celsius, recorded and considered as indication for antifungal activity. Procedure for well diffusion method. We need sterile antibacterial disc and also sterile bacterial. Sterile disc is placed in the sterile petri plate. Then the plant extract is taken in known volume by pipetting, and then the plant extract is added over the disc for impregnation to occur. In this method, if plant extract is added into this, then the disc will absorb the extract and then it is placed onto the media along with the culture to screen for antimicrobial activity. After that, in inoculated sterile separate extracts agar media, 0.2 ml of the spoon solution is prepared and spread all over the plate by spread plate method. Then the sterile disc in which we have inoculated plant extract, it is taken and placed in the center of the media by giving a gentle press, and it is kept for 4 to 5 days for the microorganisms to grow. Here are results for phytochemical investigations and also for antimicrobial and antifungal activities. The results are given in the table, and the zone of innervation are measured with the help of scale. Conclusion: the present study showed impressive preliminary phytochemical constituents in ethyl acetate peel extracts of Tunica granatum L, that is pomegranate. The results obtained from antimicrobial activity of pomegranate was that it can be used as an antimicrobial agent in pharmaceuticals. There is a wide body of scientific study and evidence to show that pomegranate has great potential in the treatment of many microbial diseases. Thank you.
Thank you so much, Sanika. Next, I have Ms. Navya Shetty presenting her topic on isolation of lipase-producing bacteria from milk and its use in oil spill cleaning.
Warm regards to one and all present. Myself, Navya Shetty, and today I'll be giving a presentation on my research topic, which is isolation of lipase, enzyme-producing bacteria from milk, and its use in oil spill cleaning. Before conducting a research, it's very important to know on what we are conducting a research and why we are conducting a research. So now, let us first see what is lipase. Lipase is a family of fat hydrolyzing enzymes. It breaks down triglycerides into free fatty acids and glycerol. This process is called lipolysis. Now, lipolysis can be brought about by two kinds of lipases. One, endogenous milk lipoprotein lipases and bacterial lipases that are produced by bacterial species like Pseudomonas, which are present in milk. Now let us see what is role of lipases in oil spill cleaning. Lipases or triacylglycerol hydrolases deal with degradation of petroleum hydrocarbons. Organisms producing these enzymes can be employed in the bioremediation of hydrocarbon. What are the advantages of using microbial lipases? Lipases from microbes are of importance due to their high development specificity of their catalytic activity, low production cost, and ease of genetic manipulation. Different bacterial species, especially Pseudomonas, are applied in biodegradation of crude oil. Now let us see what is the purpose of this research. This research was conducted to provide an insight into role of enzymes present in milk and dairy effluents. How these enzymes that are detrimental to the quality of milk can be extremely beneficial if they are used where they are needed. Oil spill has been a problem since decades, and other alternatives of oil spill cleaning can cause harm to the environment in one way or the another. Bacterial lipases are comparatively a safer alternative and can be implemented with ease if the necessary conditions are maintained. So now let us see what was the media used and what were the tests performed to check their efficiency. Media used was tributylene agar. Its composition is given. This media was divided into two parts. To one part, glycerol was added as a lipid source, and to another part, diesel was added as a hydrocarbon source. The milk sample was divided into two parts, undiluted and diluted. Loopfuls of undiluted and diluted samples were streaked on all four Petri plates, and these plates were kept for incubation for 24 hours at 37 degrees Celsius. On second day, when these plates were observed after 24 hours of incubation period, on observing the tributylene agar plate that contained glycerol as lipid source, it was evident that lipase-producing bacteria present in undiluted milk sample was able to utilize glycerol present in the media. On observing tributylene agar plate that contained diesel as a hydrocarbon source, it was evident that the lipase-producing bacteria present in undiluted milk sample was able to utilize the diesel as a hydrocarbon source which was present in the media. Now, what is the result obtained from this test? From the test results obtained, it is evident that lipolytic enzymes present in milk produced by microorganisms are capable of breaking down crude oil hydrocarbons. These microorganisms, in absence of a lipid source, are able to use petroleum hydrocarbons as their source of food and energy. Now, the conclusion can be drawn that lipoprotein enzymes, lipases present in milk, can be of tremendous use in bioremediation of oil spill. Bacterial lipases are a better alternative at bioremediation of crude oil present in oil spills. So that was my research. Thank you. And lastly, I would like to thank my professor and my mentor, Ms. Niyati Tiwari ma'am, for guiding me throughout. Thank you so much.
Thank you so much, Navya. Now I call upon Ms. Aishwarya Kumar with her title on media optimization for PHB production by Rhizobium species.
My name is Aishwarya Kumar and today I'll be giving a brief presentation on media optimization for polyhydroxy butyrate production by Rhizobium species that are isolated from root nodules. This paper is written by Aishwarya Kumar and Shraddha Singh. We are third year students of Thakur Shamnaran Degree College, and it was the research was done under the guidance of Niyati Tiwari ma'am. And to begin with, we polyhydroxybutyrate (PHB) is basically a biopolymer that is produced by microorganisms as a secondary metabolite. It is currently being investigated or a candidate of high potential to substitute petrochemical based polymers like polythene and polypropylene that are being used as plastics right now. And the reason why it is that it is a polyester of the PHA family, but the properties of it, like its biocompatibility or how it is non-toxic or how it leaves non-toxic residues, the favorable mechanical properties that makes it a good candidate for becoming bioplastic, its amenability or how readily it biodegrades in biologically active environments, and its similar properties to polypropylene or current plastics is what actually makes this a great candidate for to be a bioplastic alternative, and which is why today we are researching about it. As the name suggests, it's a biopolymer, and so microorganisms are the source of PHB. It is produced as a secondary metabolite in microbial metabolism when the microorganism is living in unfavorable or stressful conditions. The two most important conditions to focus on here is that there is excess of carbon source in the environment and a nutrient stress created on the nitrogen and phosphorus and sulfur sources. This is an intracellular compound that serves as an energy reserve, is used for energy storage, and since it is in an amorphous liquid granular lipid form, it survives well under any kind of stress, and which is why nitrogen fixing bacteria or rhizoperic microbes are great PHB producers. Is that rhizoperic soil makes a great habitat for these microorganisms. The reason why rhizoperic soil makes an excellent habitat is that this soil has constant fluctuations in nutrient concentration due to plant root activity, and these microorganisms survive well in these changing conditions, and PHB production serves as a competitive advantage with in comparison to the other microbes that are present there. So today we are going to discuss about this experiment in which we collected root nodules and a culture suspension was made for isolating nitrogen fixing bacteria or rhizobium species in it. We isolated them and screened them using Sudan black stain for PHB production. When these strains were found to be positive for PHB production, they were used as test culture to optimize media specifically for that isolated rhizobium species. Talking about isolation and screening, root nodules were plucked off out of unwashed roots of methi, which is a legume which has a nitrogen fixing bacteria. These root nodules were used to create a suspension of which the undiluted sample, the 10 raised to minus 2 and 10 raised to minus 5 dilutions, were plated on nutrient agar plates and Yema plates with Congo red in it, and they were incubated at 37 degrees Celsius for 48 hours. After 48 hours, it was observed that all the plates showed growth on them, and when stained with Sudan B black stain for PHB production activity, all the plates retained black stain, which indicated that the culture suspension we had was positive for PHB production, and this gave us a further check for using that as a test organism for media optimization studies. Coming to media optimization, we had an original unmodified composition in the table given in front of us, and in this composition mannitol was the original carbon source and yeast extract was the nitrogen source. 50 ml of this yeast extract mannitol broth was used in 12 flasks to study specifically 3 aspects of media optimization: effect of pH, effect of nitrogen source concentration, and effect of type of carbon source that is used. All the 12 flasks were inoculated with the culture that we isolated and screened for PHB production previously, and then were incubated at room temperature in a rotary shaker for 48 hours. After incubation, flasks were observed for a type of white spherical growth that you can see in the picture. The white spherical pellets were observed. These pellets were what we were looking for, and hence they were filtered out of the broth and dried and weighed, and then dry cell mass was calculated, and the results and analysis of that calculation of dry cell weight is discussed further. Firstly, as said, effect of pH condition was studied, and for this isolate, even though most PHB producers grow from anywhere between pH 4 to pH 11 range. For this isolate, only pH 7 or the neutral condition showed growth, making it the optimum pH for rhizobium species, and the alkaline with pH 9 and acidic condition with pH 5 had no growth observed in them. Moving ahead to effect of nitrogen source concentration, the graph in front of us represents all the data that was collected. As you can see, 0.3% had no growth seen in it, while the rest three concentrations showed growth, but even though we are looking for a minimal nitrogen concentration, extremely low amount of nitrogen source also was not ideal for growth of the cells. Most growth was seen at 0.1 concentration, followed by a gradual decrease in the 0.2 percent concentration, which indicates that the optimum concentration is between 0.1% to 0.2, making it the optimum range. And to further exact studies for nitrogen source concentration should be carried out in the optimum rate suggestively, and for this isolate 0.1% would be the optimum nitrogen source concentration, or would be near to it. Effect of type of carbon source. In this, we were able to study 5 different types of carbon source: cornstarch, jaggery, sucrose, glucose or dextrose, and mannitol. Mannitol was the original unmodified component of the media, while cornstarch and jaggery are two agriculturally sourced carbon sources, and sucrose and glucose were chemically pure sugars. Starch was not suitable due to its turbid, viscous consistency, which made it very difficult to filter out any solid that was formed in the broth, while there was constant sticky starch deposition in the form of clumps, which made it indistinguishable if there were any pellets formed. Jaggery and sucrose and dextrose all showed growth, of which sucrose was the carbon source that gave the highest yield, followed by mannitol and then glucose. Jaggery, while jaggery also being a natural sucrose source, gave a better yield than glucose and mannitol. Due to some impurities or if a presence of other components, it gave a subpar yield to the chemically pure sucrose. To conclude, the main aim of the study was to optimize media for production of PHP, specifically for this nitrogen fixing strain, and we studied 3 aspects of it: pH, nitrogen source concentration, and carbon. The isolated PHP producing strain we found out grows optimally at neutral pH and when nitrogen source is around 0.1% in the media, and it majorly favors sucrose as its carbon source. There are obviously many possibilities ahead for more research options for more economic and ecological production of PHP to be further used as bioplastic, but this proves that the study is important as it has many applications in fields and a lot of future prospects. I thank everyone for listening to me. I thank Niyati ma'am for guiding us through this research, and I thank the Department of Microbiology ID and all the faculty for their cooperation, and the college for providing us laboratory facilities to carry out this research. I would like to end my presentation here today. Thank you for listening.
Thank you, Aisha. Next, I have Ms. Kanak Rajendra Kumar presenting her title on effect of body pains on normal flora.
Okay, so greetings to one and all present here. I am Kanak from SYBSP ID Microbiology, and today I am here to explain my topic, effect of body pains on normal flora.
So, introduction. We all are aware about normal flora, what they are. They are like microbes which live on other living organisms or other inanimate objects without causing any harm to them. So they are like they defend against pathogens, educate the immune system, helps in wound healing, moderates progression of diseases, and in return they receive some nutrients. So we all know that skin is the initial physical barrier, and it's cool, acidic, dry, and is harsh for most of the microbes. And only few types of microbes who have adapted to these conditions can successfully colonize there. Okay, so the majority is like they obtain the nutrients from sweat, sebum, dead skin cells by using these proteases and lipases. And a single square centimeter, a very small area of the skin also contains a lot of billions of microbes, which can be like bacteria, any bacteria, viruses, protozoa, fungi, mites. Okay, so about body paints, they are basically to decorate our skin, the outermost surface, and like the use of body paint in the visual arts is essentially limited to the forms of avant-garde art, like innovating art, something exploring something new. The methodology. Next is the methodology, how we performed it. First, we did serial dilutions of the body paints. Then we impregnated the dilutions on Whatman paper discs in order to prepare the discs. Then we let those discs be in the solution for around 10 minutes. After that, we launch the sample of the normal flora of skin on the nutrient agar media plate. Then septically we place those discs on the launch sample and then incubate that thing for 24 hours at 37 degrees Celsius in order to study whether the body paint is toxic against normal flora or not. So here's what the results we got. There was a normal growth seen. Also, more number of colonies were observed on the second day, like on the day of the result, like around the body paint impregnated disc. So the conclusions drawn out from this were the body paints were found to be non-toxic against the normal flora of the skin. This concludes that they have no effect on the normal flora. So I would like to I'm extremely grateful to our principal, Dr. Sharat Fulari sir, our coordinator of the Department of Microbiology, Udayman Yadav sir, and our assistant professor, Mrs. Niyati Tiwari, for their guidance and support. Thank you so much.
Thank you, Kana. And now I have Ms. Shraddha Singh, whose topic is isolation, screening, and extraction of PHP producing bacteria from soil sample.
Good afternoon, teachers and all my dear friends. Myself Shraddha Singh, and today's topic is isolation, screening, and extraction of a PHP producing bacteria from soil sample. As you can see, there is a table of content that which you're gonna see this throughout the presentation. That is abstract, introduction, materials and method, result and analysis, acknowledgement, and bibliography. So bioplastics are naturally occurring biodegradable polymers which is made from polyhydroxyalkanoates, that is PHA, in which poly-3-hydroxybutyric acid PHB is the most common. PHB serve as energy storage molecules and accumulate intracellularly as storage granules in microbes. PHB can be used as an effective thermoplastic and has many characteristics similar to those of the standard commercial plastics like polypropylene. PHB-based plastic substitutes are less flexible than traditional plastics. They are completely biodegradable and leave behind no residue. This work utilizes the biodiversity of bacteria of isolates various specific from different environments, and screen them from their ability to produce PHB. PHBs are energy reserves synthesized by different microorganisms such as alkegenes, Pseudomonas, Staphylococcus algae, and excess of carbon and limitation of nutrients like nitrogens. Petroleum-derived plastic that is commercially available have become an integral part of our modern life, which successfully replacing various other products over the years. From automobiles to medicine, plastics are utilized in almost every manufacturing industry in the world. Approximately 25 million tons of plastics are produced by the plastic industry every year. The constituent dependency on commercial non-biodegradable plastic cause an adverse impact on environment, crude oil reduction and pollution. Disposal of plastic wrap waste through the incisions may generate toxic product and is expensive. Thermal process and recycling can be done, but it is time consuming. Process and alternations may occur in the plastic with the time. So to overcome the hazardous impact of commercial plastic, there is an alternate of the biopolymers such as aliphatic polyester, polyactides and polyhydroxyalconate and polyhydroburate. PHB has been the most promising biodegradable plastic as an alternative to petrochemical plastics. This is due to their biocompatibility, biodegradability and versatile properties make it eco-friendly substitute for synthetic polymers. The molecular weight varies from 223 into 103 K Dalton. The weight was depending on the species that actually producing the polymer. PHB has more advantages because it is far less permeable than PE and PP. This is known as better material for food packaging, need less to use antioxidant. Some industrial applications of PHB has been hampered owing to its low thermal stabilities, and industrial application of PHB has been hampered. Excessive bitterness upon storage. The main fermentation strategies used to obtain bioproduct are batch culture, fed batch culture, continuous culture and two-stage fermentation. The materials and method. The first we did is sample collection isolation of a pure culture. We collected soil sample in a sterile bottle from a ground. Then there was a screening of the PHB producing isolate by Sudan black bee staining. Then there was a characterization of PHB producing isolates, in which we identified and categorized by morphological and biochemical characterization according to the Burgee Manual of Determinative Bacteriology. Then the result and discussion came that isolation of microorganisms first. Microorganisms were isolated from soil sample and independent colonies were obtained by serial dilution. A total of 10 bacterial colonies with different morphological features were selected and the number were given to each colony. These colonies were isolated on Newton agar plate and Yema plate. These are the Newton agar plate and Yema plate in which isolated colonies are obtained. Screening of the PHB producing bacteria. Among these 10 colonies, 3 colonies showed positive for Sudan black bee staining. These 3 colonies are 1, 2 and 3. They were named as strain 1, strain 2 and strain 3 are showing high color intensity with Sudan black bee, and the microscopic pictures are depicted in figure 2. Here are strain 1, strain 2, strain 3 and strain 4, in which there is a showing of PHB granules with Sudan black bee staining. The characterization of PHB producing isolates, the morphological and biochemical characterization were done, in which the morphological characteristic, the strain 1 has bacillus in shape, the reddish pink in color and gram positive in gram staining. Strain 2 were bacillus in shape, reddish pink again and gram positive. Strain 3 was bacillus in shape but purple in color and were gram negative. And strain 4 was bacillus in shape, purple in color and gram negative in gram strain 4. Then in biochemical test, strain were negative for indole production, positive for citrate utilization and positive for catalase. Strain 2 were positive for indole production, negative for citrate and catalase. Strain 3 were positive for indole and catalase but negative for citrate utilization. And strain 4 were negative for indole and citrate utilization but positive for catalase. Over here there is a picture of indole production and citrate utilization. They concluded the main aim of this present study was to isolate the PHB producing bacteria from soil sample. Nowadays, researchers are focusing on biopolymer producing microorganisms for developing biodegradable plastic. The medium used for the PHB isolates was simple medium, are less cost effective, and PHB yield from these isolates were high compared with the earlier reports. Among all the isolates, the strain 1 is showing more production of biopolymers, and the PHB produced from its strain will further be characterized by analytical techniques like infrared species and gas chromatography analysis. This author is indeed grateful to Dr. Sharat Fulari, Principal of Zavadu Singh Charitable Trust, Thakur Shamna Degree College, and the management of the college to grant me the permission to use infrastructural facilities, and also for moral boosting, cooperation and support throughout the tenure of the project. I also wish to extend my heartfelt gratitude towards Mrs. Niyati Tiwari ma'am, Bharti ma'am, Sonali ma'am, Kunal sir, Uday sir, Aishwarya ma'am for their timeless support and cooperation, without which completion of this research project was not possible. Thank you.
Thank you so much, Shraddha. And now I call upon Mr. Kunal Thakur, whose topic is to study the effect of lipase enzyme extracted from intestine microflora of animal and bird gut on margarine.
Good morning, everyone. Myself, Professor Kunal S. Thakur, Assistant Professor, ZSCTS Thakur Shamna Degree College, Kandivali, Mumbai. Today, I am presenting my paper on the topic of extraction of lipase enzyme from animals and birds' fat and its activity on margarine. Now, abstract of the topic is that lipase are extremely adaptable enzymes that have caught of interest of numerous industrial processes like animal, vegetable, and microbial source of lipase are all possible. Long chains of triglycerides are hydrolyzed by microbial lipase. The lipase enzyme microbial origins are theoretically flexible and capable, and so they are widely used in industrial use of the production of many molecules. The hydrolysis, esterification, and alcoholysis reactions all are catalyzed by special lipase enzymes. So these enzymes are used from immemorial time, and the immobilization method and carrier type affect the immobilized enzyme when choosing a carrier factor including biocompatibility, chemical and thermostability, insolubility during reactions, ease of rejuvenation and reusability. So we can use this enzyme for many processes and for synthesis of many compounds like biodiesels, food, beverages, leather, textile, detergents, etc. So our research based on estimation of the quantity of the lipase enzyme present in intestine of the chickens and goat. Now, what is lipase enzyme? Lipase enzyme consists of many molecules, many subordinates, which are useful for the degradation of the lipids, which are desirable by the many industry. So, in comparison to lipids derived from plant and animals, microbial lipids are more desirable. But we are not getting a successful lipase from the same sources. So what we needed? We needed a special lipase enzyme derived from the goat or hen's source. The bacterial isolates offer higher activity such as neutral and alkaline pH optima and thermostability associated with the yeast. Bacterial strains such as Pseudomonas, Bacillus are known to produce some special type of lipase enzyme, And for this reason, in this research, we have used the intestinal microflora of the birds and animals to get some special microorganisms, and from those sources, we have extracted our lipase enzyme. Now, the market is expanding because microbial lipase have greater advantage over animal and plant lipase. So, we are using this enzyme for many other processes like extraction of many stability molecules, some production molecules, and this is very important to get the lipase enzyme from microbial source. These have broad application in variety of industry like cleaning, biofuel, pharmaceutical, textile, cosmetics, etc. Now, there are several cutting edge purification techniques available in the market, but in this process, we are extracting the cells from different sources like like in the sternal of the birds and the chickens. So we extracted the cells from that environment and growth into the culture broth, and the enzymes were extracted using different method like centrifugation and filtering method, and these then estimated with the help of potassium iodide titration method. Now, here is the method and requirement. In this method, we just sample collected from different cultures like mutton and chickens. The fats were kept for the 1 week in the allow of the microorganism to grow in there. After that, we have cultured that microorganism on the two different plates, and then after that, with we extracted the organism on nutrient agar plate and enrichment method. After that, we have extracted the lipase enzyme using NaOH and lysis method and rotation at centrifugation of 6000 rpm. And after extraction of the enzyme, we just do biometric analysis using potassium iodide and sodium thiosulfate. So after that, we got our enzyme in different formula using volume of sulfate formula like S into N into 1000 PV value. We calculated the weight of the lipid present in the intestinal microflora of the selected organism. Now, this is the observation table, what we got. Here we can see the animal source show high OD value than bird source. That means the enzyme present in animal gut flora is much more useful to clarification of the lipid concerns. Now, this is the comparison between the two sources where we can see that animal lipase has more activity based as compared to bird lipase. This is the percentage bar graph which show that nearly animal lipase has twice the activity as compared to bird lipase enzyme. And this is the result of the experiment that more amount of lipase was extracted from the animal source as compared to the bird sources. Now, this is my final conclusion that in comparison of the lipids derived from the plant and animals, microbial lipids are more desirable because of the wide range of the catalytic actions that can perform what we see in our experiment. And in comparison between the animals and the birds, animals show more lipase enzyme as compared to the bird source. Okay, thank you. This is the references what we used in our experiment.
Thank you so much, Kunal sir. I have Ms. Akanksha Dubey, whose topic is to study the effect of auxin on different microflora.
Greetings to all the people present here. My name is Akanksha Dubey. I am here to present on the topic effect of auxin on bacteria under the guidance of Mr. Udayban sir and Mr. Kunal Thakur sir. Abstract: auxin is a growth hormone found in plants. It's used by plants for growing. It has antimicrobial properties found in young plants for growing. Various bacteria promote plant growth in the rhizosphere as a measure of securing and enlarging their ecological niche. These interactions are mediated by plant growth regulators such as auxins and indole acetic acid is one of the physiological active auxins. In this study, we isolated an usual bacterial strain from food processes waste with high efficiency and demonstrated its effect on plants rooting and early stage growth. Keywords: auxin, antimicrobial properties, its effect on bacteria. Introduction: Matki. Moth beans or matki is a staple legume in various cuisines across India, often consumed either as a sprout or in the cooked form. Quite popular in Maharashtrian cuisines, moth beans are also known as mat bean, dude bean, or Turkish bran. These tiny beans oblong in shape, available in brown, reddish brown, and green colors, are rich in protein and go with the botanical name Vigna actiflora. Moong. Moong bean is an important pulse consumed all over the world, especially in Asian countries, and has a long history of usage as traditional medicine. It has been known to be an excellent source of protein, dietary fiber, minerals, vitamins, and significant amount of bioactive compounds, including polyphenol, polysaccharides, and peptides. Therefore, becoming of bioactive compounds, including polyphenols, polysaccharide, and peptides. Chana. Chana is a cool season legume crop grown worldwide as a food crop. The seed is main edible part of the plant. It is also called gansogram or Bengal. It ranked third among the food legumes after beans and peas. More than 50 countries are reported to grow chana. 22 cultivate more than 20,000. Material and method. Sample: day 1, moong, chana and matki were soaked overnight and then removed it from water and tied with muslin cloth. Day 2, extracted the germinated root from the legumes. Then it was pasted in mortar pestle, transferred it in 3 different dilution tubes. Requirements: micropipette, chana, moong, matki, sterile agar plate, chana, culture E. coli and azoles, mortar pestle, and cough borer. Agar diffusion well method. A cough borer is also used to punch holes on an agar plate and used to perform well diffusion assay in microbiology to study bioactivity. Agar well diffusion method is widely used to evaluate the antimicrobial activity of microbial enzymes. Results: after incubation, zone of inhibition was observed around the well of each sample. Auxin showed visible zone of inhibition in both the agar plates under the used bacteria E. coli and azoles. These are the pictures showing the result. Conclusion: we conclude that auxin extracted from chana gives positive result against azoles, whereas the auxin extracted from moong gives positive result against E. coli. Thank you.
Thank you so much, Akanksha. Now, I call upon Ms. Sakshi Karekar, whose topic is to study the configuration and advantages of these graphs.
Warm greetings to everyone. Myself Sakshi Karekar from third year microbiology department from ZSCB Shamnar Degree College. I'm going to present on the research topic configuration and advantages of beeswax under the guidance of Professor Nirvani others, coordinator of microbiology. Beans produce wax. Bees need wax as construction material for their clothes. They may use it in their wax glands, which are fully developed 12 to 18 days morphers. In order to reach the wax glands stimulation activity, however, in emergency situations, wax releases can be deactivated. Latest case of wax are used during the growth phase of bee colonies under moderate climate conditions during April and June. A Bibliography on the synthesis of beeswax is given in the monograph of Hepburn 15. The main raw materials for wax formation are carbohydrates, that is the honey, sugars, glucose, and glucose. The ratio of sugar to wax can vary from 3 to 30. A ratio of around 21 being typical of Central Europe. The stronger the polyene, the smaller the ratio. The more economical the wax production for the polyene. One landstrom frame containing 100 grams of wax can put 2 to 4 kgs of honey. Manufacturing methods for beeswax. Hot water extraction using forced immersion. The combs are placed in a tightly tied sack. Place sacks in a receiver filled with water and water. As wax is lighter than water, it will filter through the tube and rinse to the surface. After all combs have all melted, let the pot cool. The wax solidifies as it forming a block on the water surface. Throughout paste left in the sack, extraction with boiling water and a wax placed a 120-liter container with 20 to 30 liters of boiling water and left to it. When all the wax has melted, remove the firing and dip the contents into a tube and press and start pressing. Combined steam and press extraction. A metal basket of old combs is plugged into a tap of boiling water, hosed with a watertight cup. A piston capable of exerting up to 15 of pressure presses the combs. The tank is kept simmering for about 1 hour. Wax runs to the top of the tank. Steam extraction combs with frame are placed into a container when vapor is introduced. The tester is steamed. Wax flows into the lower part of the container and can be collected. There are different commercial devices. Centrifugal extraction combs are wetted in boiling water and boiling mixture is put into baskets of a centrifugal wax extractor. Spinning at more than 1500 RPM. Kept at temperatures over 65 degrees Celsius to prevent the wax from setting. Pure wax runs out through an opening from the extractor. Method used for bigger manufacturing units due to expensive installation. Heat extraction with electric elements, press comb or frames between two electrically heated metal plates. Plates are pushed together. The wax melting vapor is liquid. Properties and composition. Beeswax of different varieties. Publications on the physical constant of the cone waxes of Asians and European waxes first appeared a century ago. It was soon shown the carbon chain length was on average shorter in the Asian beeswaxes than in the Penifera, which explains the lower melting points of the bond. The Asian waxes are more similar to one another than to the Penifera. In Asian beeswaxes, the amount of C31 and C33 in the pool of mineral fatty acids are reduced, but C285 hydrocarbons are increased compared to that of Mellifera. The major compounds families in beeswax are alkanes, alkenes, fatty acids, monoesters, diesters, and hydroxymonoesters, while fatty alcohols and hydroxydiesters are minor constituents. There are notable species-specific differences in the beeswax among honeybee species, but all share a complex mixture of homologous neutral lipids. The wax produced by different species of Apis mellifera are also an African adenosine wax have the same composition, but components are in different proportions, which are 3 to 7. The waxes of Asian bees, Apis florea, Apis dorsata, and Apis cerana are called Keda. The composition of wax from Asian honeybee species is much simpler and contains fewer compounds in different proportions. The different Gedda waxes resemble each other much more than any of them the mellifera waxes. Thus, Gedda waxes cannot be used as substitute of Apis mellifera wax. Gedda waxes from the Asian honeybee species are described as softer and more plastic, but do not have significantly different melting point. Properties of waxes are clear yellow to yellow brown odor, heat wax odor should be pleasant or honey-like. Chewing test, wax should not stick to teeth. Breakage test, upon breaking should have fine granular, blunt or crystalline structure. Cutting test, wax should not stick to the knife. Splitters, scratch wax with nail or knife. Splinters should have a spiral. Kneading for 10 minutes, wax should be plastic. Consistency should not stick upon cutting. Conclusion, wax is widely used as a popular ingredient in cosmetics, aromatic candles, lip balm, and lotions since ancient times. The natural products has its own benefits in our day-to-day life. Thank
Next, we have Mr. Kevat Modi, who's presenting his topic on confirmation of the location of bacteriocin expressing marker on a plasmid of Lactobacillus rhamnosus L43.
Hello, I'm Kevat Modi from Aditi Sharma Degree College, by microbiology. I want to present my topic on confirmation of location of bacterial on plasmid of Lactobacillus rhamnosus L43 under Dr. Lactobacillus rhamnosus L43 is isolated from marine water. Lactobacillus isolate was sequence which shown high similarity with the reference gene Lactobacillus rhamnosus led by SSN number EC012630. These isolates indicated the potent strain produced bacterial. Plasmid curing has been carried out for mapping. It is indicated the potent strains produced bacterial. Plasmid curing has been carried out for mapping bacteria in producing genes and plasmid DNA of Lactobacillus rhamnosus. Cells growing from protein about 10 mg per concentration ethereum bromide was selected to observe the effects of curing. In this study, have demonstrated plasmid-mediated bacterial infection. Antibiotic-resistant immunity could be lost as the curing of plasmid-bearing lactic acid bacteria. The plasmids of both gram-negative and gram-positive bacteria are involved in metabolic activity. Membrane protein producing the establishment of progressive infection and resistance. Many successful attempts have been made to isolate and sequence the genes of plasmid from many different species, including E. coli, Staphylococcus, Streptococcus, Salmonella, Listeria, and lactic acid bacteria species. The first plasmid isolation was carried out of lactic acid bacteria 1. Jamuna and Jeevaratham in 2004 reported that the lactic acid isolates from some traditional fermented foods, LABP and LABP, produce bacterial strains. He reported that both strains harbored multiple plasmids ranging from size 1.5 kb to more than 10 kb. He has shown that combination at Chris level, ethereum bromide and non-biotin could induce the loss of plasma DNA in these studies. He analyzed that non-cured strains of lactobacilli were resistant to ciprofloxacin, colistin, gentamicin, nalidixic acid, and streptomycin, but after curing, strains altered in its response by showing sensitivity to ciprofloxacin, gentamicin, and streptomycin. Selected isolates. Lactobacillus strain was isolated from marine water in Thirumunduvaram beach area using MRS broth at 37 degrees Celsius in 48 hours. The isolate was identified by 16S rRNA sequencing in phylogenetic analysis. Lactobacillus L43 isolate was sequenced which shown high similarity with reference strains Lactobacillus rhamnosus ZY excessive number KC01263 Antibiotic sensitivity test. Antibiogram of selected industrial isolate Lactobacillus carried out using Kobigo's method as proposed by Kelsea and Kumar. The isolate Lactobacillus analysis was inoculated into MRS broth, individually incubated at 24 hours. About 20 ml of MRS agar was selected with the cultures of selected isolate B24. Mixed well, poured into sterile petri plate and allowed to solidify. Antibiotic discs were placed upside down or pressed on top of the agar plates. The plates were incubated at 37 degrees Celsius overnight. Resistance was determined in terms of zone of growth around the discs. Confirmation of location of bacteriocin expressing markers from plasmid curing. Confirmation of plasmid curing nature of gene producing plasmid was determined by plasmid curing. The curing of plasmid has been carried out by treating the active intact cells of Lactobacillus rhamnosus L43 using Ethereum bromide. The strains of Lactobacillus L43 was grown to late or logarithmic phase of growth and MRS growth. The cells were harvested and washed twice with sterile 0.9% NaCl solution and 2, 4, 6, 8 and 10 milligram per ml of ethylium bromide were added to 2 ml of each cell suspension. The mixture was kept and incubator for 30 minutes respectively. The treated cells were incubated in 10 ml MRS broth, washed twice, and resuspended in 0.9% NaCl solution. After serial dilution, it was spread on MRS agar plate and incubated at 30 degrees Celsius for 48 hours. After incubation, the number of colonies was counted and stored for further study. Confirmation of curing by antibiotic sensitivity test and antimicrobial activity. Antibiotic sensitivity test was carried out as described previously. Both cured and uncured cultures were used to determine the pattern of antibiotic sensitivity. The bacteriocin was produced from both cured and uncured cultures, and antimicrobial activity of this bacteriocin was determined described previously. In both tests, the response of both cultures was correlated with each other. The presence of plasmid-encoded markers was confirmed by observing the loss of respective activity from cured cultures when compared to the response of uncured cultures. Uh, Resultant discussion. Confirmation of location of bacteriocin expressing markers in plasma. Plasmid curing has been carried out for mapping bacteriocin producing genes on plasmid DNA of Lactobacillus rhamnosus L43. As per survival curve, it was observed that ethylium bromide is found to be determinant for growth of Lactobacillus rhamnosus L43. The LD50 was found to be 10 milligram per ml. Cells growing on nutrient medium about 10 milligram per ml concentration of ethylium bromide was selected to observe the effect of cure. Table 1 and Figure 1 represent the survival curve. Obtained by ethylium bromide concentrations. In our studies, the strategy of ethylium bromide cure, could cure the plasmid of Lactobacillus species, similar finding as reported by Karthikeyan and Santosh. Antibiotic sensitivity test after plasmid curing. Cells cured of Lactobacillus rhamnosus L43 has shown loss of resistance to antibiotics, that is, streptomycin, gentamicin, erythromycin, rifampicin, vancomycin, fluorescent nalidixic acid in cell as compared to uncured cells. Table 2 represents the comparative antibiogram of Lactobacillus thalamus L43 after plasmid curing. Antibacterial activity after plasmid curing. Table 3 in particular represents the antibacterial activity of the strain Lactobacillus thalamus L43 after plasmid curing. In both tests, the response of both curves was correlated with each other. The presence, the presence of plasmid encoded markers was confirmed by observing loss of respective activity from cord cultures when compared to response from cured culture. Presently, we have demonstrated the plasmid-mediated bacterial producing antibiotic resistance and immunity could be lost as the curing of plasmids. Moreover, it has been recently reported by Pathogen and Synthesis that the Lactobacillus plantarum strains are producing bacteriocin about 2.5 kilodalton, which is plasmid equivalent. Glory in 1999 has reported that broth of antimicrobial compound producing an immunity determinants were encoded by an 0.8 kb plasmid in a L. strain of vegetable origin. Uh, while there was a plasmid associated for producing of bacteriocin of in a lactobacillus strain without affecting the immunity, indicating uh, the possibility of the immunity change of the chromosomes. Similarly, a study reports plasmid curing, which was performed by Tolinaki at the Institute of Molecular Genetics and Genetic Engineering University of Belgrade, Serbia. Similarly, plasmid profile of Lactobacillus species, as reported earlier, photocrit 2 has shown the presence of plasmid of Lactobacillus rhamnosus L43. Conclusion: according to the present study, Lactobacillus rhamnosus L43 is a plasmid-bearing bacteria which is having antibacterial activity in bacteriocin-producing markers available on the selective lysine. Thanks so much.
Thank you, Favor. Now, I call upon Mr. Sumit Sanjay Gupta, whose topic is on microbial analysis of polluted and unpolluted soil.
Hello everyone, this is Sumit Gupta from first year microbiology department from Thakur Shamnarayan Degree College. I am working under research of Sonali Joshi ma'am on the topic microbial analysis of polluted and unpolluted soil. Aim: microbial analysis of polluted and unpolluted soil. Requirement: 200 ml of nutrient agar, sterilized petri plate, sterilized 1 ml pipette, 50 grams of polluted and unpolluted soil sample, 100 ml of distilled water. Procedure: the soil sample are dried in the hot air oven and then ground to remove the clamp. Then the sample are shifted through the sieve of the different mesh shaped. 200 ml of nutrient agar was made from 5.6 gram of Na was dissolved in the 200 ml of the distilled water. The medium was sterilized using the autoclave at 121 degrees Celsius for 15 minutes. After the sterilizing process, the medium was allowed to cool down and then it was transferred into the petri plate under the sterilized condition. The soil sample was used using the dilution method. 10 raised to 4 and 10 raised to 5 of the polluted and the unpolluted soil was used during the research. The distilled water used as the diluent. The diluted soil sample of both polluted and unpolluted of 10 raised to 4 and 10 raised to 5 dilution was pipetted out using the 1 ml of the pipette. 1 ml of soil sample of both polluted and unpolluted was spread on the petri plate containing the Na media. The sample was then spread using the alcohol swab. Then the petri plate was incubated at T 37 degrees Celsius for 24 hours. Discussion of result. For polluted soil, during the observation, 10 raised to 4 soil sample showed less growth of the organism, that is more than 200 colonies. 10 raised to 5 soil sample showed very less growth of the organism, that is more than 150 colonies. Discussion of result. For unpolluted soil, during the observation, 10 raised to 4 soil sample showed very denser growth of the organism, that is less than 500 colonies. 10 raised to 5 soil sample showed very denser growth of the organism, that is lesser than 300 colonies. Conclusion. The polluted soil contains the very less amount of the organism and pollutant, whereas the unpolluted soil contains the very denser amount of the organism and the pollutant. Thank you.
Thank you so much, Sumit. Now I call upon Kevrat Modi once again, who is presenting his topic on preparation of biofertilizer and a comparative study with the chemical fertilizers.
Hello, I'm Kivut Modi from Thakur Shamnaran Degree College from Department of Microbiology, third year. I'm presenting my paper under Mrs. Niyati Pandey, Assistant Professor at ZSCT's Thakur Shamnaran Degree College. My topic is preparation of biofertilizer and a comparative study with the chemical fertilizer. Abstract. Nutritionally, fruit trees are an excellent source. In order to make biofertilizer, it is used chemically. Chemical fertilizers are bad for humans, animals, and plants alike. We ought to refrain from using chemical fertilizers. In order to determine which fertilizers produce the best results, the comparison of chemical and biological fertilizers is done. Introduction. Biofertilizers, which are created from natural ingredients and can promote the health and growth of crop plants, have been developed for many years. The need for biofertilizers is anticipated to expand because to because of the rising worldwide concerns about pollution, greenhouse gas buildup, and the increased need for plant-based foods. In this study, we provide a succinct summary of the tools available and take into account several strategies and approaches that can provide data on novel advantages features in biofertilizers. We also talk about creative ideas regarding where and how to find effective new biofertilizers. Chemical fertilizers are crucial in ensuring that the world population's rising food needs to be met. To produce crops with the highest yield possible, commercial fertilizers of these three main types, nitrogen, phosphate, and potassium, are employed. However, the increased agricultural use of chemical fertilizers causes harmful impacts on ecosystems. The pharmacological significant compounds present in medicinal plants attract scientists. For the prevention of disease, citrus fruits include a number of healing ingredients. Therefore, tests were conducted to determine the antibacterial efficacy of peel extracts from a variety of locally accessible citrus fruits. Antimicrobial properties of biofertilizers: the secret to plant and human health may lie in microbial diversity. However, it is unclear how to improve microbial variety, which is important for health reasons. Plants themselves have a distinct microbiome, and it is determined by how well it functions. Different parts of the plants have a different plant microbiomes. For instance, the rhizosphere is responsible, reasonably stable and protected interface to the surrounding soil, and is rich in nutrients from root exudates. The phyllosphere, which solubilizes the air plant interface, it lacks in nutrients and has a more dynamic environment that is influenced by antibiotic influence from the outside environment. However, the endosphere links the two in microenvironments and the microorganisms that live there. Preparation of biofertilizers. Step 1. Fruit peels are cut into pieces and gathered in a dish. Step 2. Fruit peels are dried for 24 hours at 70 degrees Celsius in an incubator. Step 3. Fruit peels are been powdered and after dehydrating and grinding. Step 4. The soil used to plant the fenugreek seeds was then combined with the powdered biofertilizer. Step 5. The seeds were then stored for a week to grow in a proper circumstance. Growth with chemical fertilizer. First, the dirt was blended with the chemical fertilizer. Step 2. In the soil mixture, fenugreek seeds were allowed to develop for a week. Step 3. Both potted fenugreek seeds underwent computed and observed growth. Observation: in both pots there was a visible plant growth. Comparing the two, the one with the biofertilizer exhibited a superior rate of vegetative development. Because there are ample of natural sources of oxygen, nitrogen, and phosphorus, as well as many other nutrients, the plants that were used for biofertilizer grew higher. In contrast, the leaves were wider and greener. The presence of mycorrhizae in the nodules made the roots system stronger. Results: chemical fertilizers have reduced soil fertility in modern agriculture, rendering it unsuitable for growing crop plants. Additionally, the extensive use of these inputs have resulted in serious environmental and health risks, including soil erosion, water contamination, pesticide poisoning, a decline in groundwater table, water logging, and the loss of biodiversity. The growing in popularity for use in crop production, biofertilizers naturally activate the soil's microorganisms while being more affordable, efficient, and environmentally friendly. They are also restored in the they also restore the soil's natural fertility while defending it against drought and soil diseases, which in turn promotes the plant growth. Further, further study and development are required to comprehend the mechanisms of action of diverse biofertilizers and to identify more capable rhizobacterial strains and carrier materials in order to ensure the success of biofertilizer technology. Thank you so much.
Thank you once again, Keval. And now I have Ms. Shani T. John, whose topic is on isolation of antibiotic-producing microbes from sediment samples from Rajakamangalam Estuary, Kanyakumari district.
Good morning. My name is Shani. I'm from Center for Marine Science and Technology, and my topic is isolation of antibiotic-producing microbes from sediment sample from Rajakamangalam Estuary, Kanyakumari district. And we all know that soil is the major reservoir of microorganisms, produces antibiotics, and the world is focusing on the production of newer antibiotics from different sources, both from terrestrial as well as marine sources. And because of the presence of antibiotic resistance among different diseases, both the human as well as animal diseases, and the more research is being emphasized on the identification of newer sources of antibiotic producing microorganisms, and therefore these soils are contributing more to the microbial world, and more emphasis is being given on this field. Therefore, I have attempted to use or identify the soil samples for potent antimicrobial producers. I have selected around 5 different sites for the isolation of microorganisms from sediment sample, which is from Rajakamangalam region, which is an estuarine region. And I have used different methodologies for screening, like after the collection of samples from the sites, I have made the isolation of this particular strains from the samples by crowded plate technique. That is, after serially diluting the samples. The samples were poured onto Petri plates and incubated for a suitable period of time. And the plates with a countable number of colonies with different morphological features, that is clear zones, were also selected. And these isolated colonies were further subcultured. Growth is purified by streaking, and it is maintained for further studies. In the screening activity, primary screening of the particular pathogens for antimicrobial activity was done by cross-streak method following the method of Shaw et al. And secondary screening was done by agar diffusion method. And after the secondary screening, the potent strains were chromatographed by TLC and column chromatography, and the active components were also again tested for its antibacterial activity as well as for minimal inhibitory concentrations. And the isolated microorganisms, in generally all these isolated microbes were also tested for its antibiotic susceptibility against the commercially available antibiotics. And the potent strain was also purified by UV and STIR analysis, and it also was sequenced to identify the particular microorganism. And here, after primary screening, the samples for the number of isolates what I have obtained from 5 different sites were like 224 strains, and most of the strains were found to be gram negative in nature, and minimum amount of strains were gram positive in nature, and again most of the strains were non-pigmented and some were pigmented. And here we can see that most of the gram negative strains were potent producers of antibiotic activity rather than gram positive strains. Based on the biochemical analysis and with the help of Burgess manual, we have predicted the presence of E. coli proteus species Klebsiella and Bacillus prosobacterium species from samples. Based on the morphological as well as biochemical nature, some around 24 strains were subjected for primary screening. And the primary screening, you can see that the strains S3, W4 and W9, which is highlighted in the table, showed potent activity against the tested human pathogens. Here we have used around 10 different human pathogens for testing the antibacterial activity. The pathogens were purchased from VIC. Then the potent strains were the strains which were found to produce potent activities were further used for secondary screening. In the secondary screening, the strains S3, W4 and W9 were used. The strains were named after its sample collection size, and these three were the potent strains which are subjected for secondary screening, and the secondary screening was done with the help of agar diffusion method. And we can see that the strain S3 was found to be more potent in inhibiting the growth of the pathogens like Shigella, Escherichia coli, Staphylococcus, Vibrio, Pseudomonas and Bacillus species. Therefore, for column chromatographic separation and all, I have used this S3 strain as the main source of agent. So the strain S3 was used for fractionation using TLC. Initially TLC was done to find out the type of fractions which can be eluted from the solvent system, where I have used a set gradient solvent system for lower order. That here I have used hexane, chloroform and acetone, where hexane acetone fractions are good enough to bring about 10 different fractions, and the 10 different fractions were also assessed for its antibacterial activity, where the fractions which is obtained from 80-20 hexane acetone column was found to inhibit or produce potent activity against the tested pathogens. The pathogens, it can inhibit the pathogens from 6 mm to 5 mm, that is like Streptococcus pneumoniae, Klebsiella pneumoniae, then Staphylococcus aureus, E. coli. All the strains were inhibited by these potent fractions. Therefore, this strain was further purified by UV and STIR analysis. Also, the fraction was also tested for its minimal inhibitory concentration, where 0.1 mg was quite enough to bring about the inhibition of the particular pathogens, where you can see that inhibitions of around 5 mm was produced by 0.1 mg of this 80-20 hexane acetone fraction against different pathogens. And the UV and FTIR analysis were also done to elucidate the or to predict the type of compound which is present in the component. Sorry. As also the antibiotic resistance pattern of the isolated strains were also tested against commercial antibiotics. Then different antibiotics were used, and you can see that the strain S3, which is being represented Photobacterium species, were resistant against most of the antibiotics. This may be because of the genetic makeup or because of the availability of this particular pathogen in the environment for a longer period of time, and maybe because of metabolic activities. And all the other strains were also, you can see that, were resistant to most of the tested antibiotics. So the compounds which is been purified by UV and FTR analysis, where the maximum peak of absorbance was observed at 220 nanometer, which shows the presence of amines and halides in a particular compound. This is the FTIR pedal. And the strain is showed potent activity against the pathogens were also tested for its for the presence of tested for the presence of a particular pathogen, where isolation of genomic DNA was carried out to obtain the pure DNA. Then 16S RNA amplification from the isolated DNA sample using PCR, and amplified product showed a fragment of 1.14 kilobase, and this amplified 16S RNA of S3 was subjected to purification and sequencing, where we have identified the particular compounds with the photobacterium tolerance, which has more similarity with the particular strain. And these are all these amplification code as well as bladder and phylogenetic tree of the particular organisms. In conclusion, I would like to say that as the resistant organism, as the disease resistance is emerging, and there is a vast need for antibiotic producers, such kinds of research has to be encouraged so that potent newer type of antimicrobial products can be isolated. Therefore, these products can be used in ease for the eradication of particular disease, both in the marine as well as human diseases, which will benefit the world in many ways. Thank you.
Thank you so much, Ms. Shani. Now, I have Ms. Neha Gupta, who is presenting her topic on antibacterial activity of different types of honey.
Hello everyone, this is Neha Gupta from first year microbiology department, and I'm from Thakur Shamnaran Degree College, and I'm working under the guidance of Dr. Bharti Wadekar ma'am, and my topic is antibacterial activity of different types of honey. These are the following points I'm gonna cover in my today's video. So let's begin with our introduction. Honey, a sweet natural product known for its nutritive value and beneficial health effect, is produced by honeybees, Apis mellifera L, which directly selects nectar from plants or excretion of plant-sucking insects, that is honey dew. Honey is essentially a concentrated aqueous solution of carbohydrates, mainly fructose 38%, second glucose 31%, third other oligo and polysaccharides consisting approximately 95% of the dry weight of honey. Apart from carbohydrates, honey contains 200 different compounds such as enzyme, example glucose, oxidase and catalysis, proteins, vitamins, example vitamin C and E, amino and organic acids, lipids, minerals and phytochemicals, example polyphenols. Honey varies greatly due to botanical and entomological source as well as climate change and environmental conditions. Honey is used in modern medicines, represents an attractive alternative treatment to combat multi-drug resistant pathogens. The antibacterial activity of honey is attributed to different factors including hydrogen peroxide, a low pH and high osmolarity, methylglycosol, MgO and the antibacterial peptide B, defensin 1 have been identified as important factor of the antibacterial activity exerted by certain types of honey. There are number of studies documenting the antibacterial activity of honey against various bacterial pathogens. Finding has suggested that further research could identify medically useful honey types with distinct advantages. Antioxidant properties of honey are also considered important. It attributed mainly to its polyphenol, example flavonoids and phenolic acids, antioxidants, enzymes, example catalase and peroxidase, vitamins, example vitamin C, Maillard reaction products, bacterial strains and growth conditions. The antibacterial activity of different types of honey was tested against Staphylococcus aureus, S. aureus strain, Pseudomonas aeruginosa, P. aeruginosa, which are procured from the college laboratory. Let's begin with the materials and methods. Honey sample. A total of 3 different types of honey which were available in market area were provided by individual beekeepers and beekeepers association. Each sample was assigned a unique reference number. Honey samples were stored in glass container at room temperature in the dark. Different brands of honey as positive controls includes Patanjali, Saffola and Zandu, whereas laboratory synthesized honey as a negative control consists of 3 gram of sucrose, 15 gram of maltose, 18.1 gram of fructose, 67 gram of glucose, 34 ml sterile deionized water. The solution was heated to 56 degrees Celsius in a water bath. Agar-well diffusion assay. The assay was performed on the basis of the Clinical and Laboratory Standards Institute bacterial culture grown in Miller-Hinton broth adjusted to 0.5 McFarland turbidity. Agar plate was inoculated with roughly 10 raised to 4 CFUs over the entire surface of the plate. Then 3 wells of 6 mm in diameter were cut into surface of the agar using a sterile cork borer. 100 microliter of the tested honey types and laboratory synthesized honey were added separately to each well. The plate was incubated at 37 degrees Celsius for 16 to 18 hours. The diameter of the inhibition zone, including the diameter of the well, was recorded. The diameter of the inhibition zone, if present in a negative control, was recorded and subtracted from the inhibition zone of the tested. Each assay was carried out in triplicate. Assessment of the total phenolic content (TPC). The total phenolic content of the honey sample was determined in accordance with a modified version of the Qualitative method. The process follows as A. A 20 microliter of sample was added to the tube containing 1 milliliter of deionized water. B. 100 microliter of polycoagulative reagent were added to the mixture and the flask was toppered and allowed to stand at room temperature for 3 minutes. C. Thereafter, 280 microliter of 25% water by weight sodium carbonate solution and 600 microliter of deionized water were added to the mixture. D. Following 1 hour of incubation at room temperature in the dark, the absorbance was measured at 765 nanometer versus a blank containing polycoagulative reagent and deionized water without the tested sample. E. The measurement of absorbance was conducted on a Hitachi U1900 radio beam spectrophotometer. F. The optical density of the sample and distilled water at 765 nanometer was also measured. The total phenolic content was determined using a standard curve of absorbance value correlated with standard concentration 50 to 1500 microgram or per milliliter of gallic acid. The results are expressed as gallic acid equivalents GES. Results: antibacterial activity of the honey type against S. aureus and P. aeruginosa. Initial screening with the agar-well diffusion assay revealed that all the tested honey types exhibited antibacterial activity against S. aureus and P. aeruginosa. In any case, the antibacterial effects exerted by the tested honey types were more potent to against S. aureus as demonstrated by larger inhibition zone compared to the effect against P. aeruginosa. Total polyphenolic content (TPC) of the honey types. The TPC of the honey samples as measured by the polycoagulative method range from 0.55 sample 1 to 0.92 milligram GAE. Discussion: 3 honey types were screened for their antibacterial activity determined. Initial screening with the agar-well diffusion assay demonstrated that all tested honey types exhibited antibacterial activity against S. aureus and P. aeruginosa. Nevertheless, the tested honey types demonstrated smaller inhibition zone of P. aeruginosa compared to those of S. aureus, thus indicating that P. aeruginosa is less susceptible to antibacterial activity of honey. This finding is in accordance with the finding of previous study possible reflecting a higher instinct. Conclusion: the present study was the first to examine antibacterial activity of honey type. The tested honey types exerted antibacterial activity against S. aureus and P. aeruginosa. Importantly, some of the honey types had a higher antibacterial activity, which is used as an alternative medicine. It seemed that hydrogen peroxide and proteinaceous compound found in a honey type were responsible, at least in part, for the observed antibacterial activity. In addition, the TPC of the honey types accounted for the antibacterial activity against P. aeruginosa. Further investigation as this may lead to application in medicines and food industry. And this is for
Thank you so much, Neha. A warm thank you to all of you for attending our conference.